বা
স্যার, প্রফেসর জানা, ডক্টর কুন্ডু and ডক্টর গৌতম দত্ত, স্যার আমাদের দিক থেকে একটুখানি আপনাদের সাজেশন লাগবে না, আমরা একটা অসুবিধাতে পড়েছি। সেটা হচ্ছে আমাদের আইডির প্রবলেমের জন্য যারা ওয়েবসাইট থেকে জয়েন করতে চাইছিলেন তারা কেউ জয়েন করতে পারছে না, মানে পুরোপুরি একটা টেকনিক্যাল ফল্ট। আর মাত্র 16 জন এখানে আছি আমরা, স্যার কি করা যায়?
তুমি যদি ওদের লিংকটা পাঠিয়ে দাও, ওরা কি ঢুকতে পারছে না?
একটা কিছু টেকনিক্যাল ফল্ট হয়েছে, আমাদের টেকনিক্যাল টিমের অতনু আছে স্যার। আসলে এই অডিয়েন্সে আপনাদের যে বলতে বলবো সেটাও মানে এত কম অডিয়েন্স, এখানে 150-র উপর অডিয়েন্স থাকে। আমাদের স্যার কি করা যায় আমরা, আমি ঠিক বুঝতে পারছি না আর কি।
না,
লিংকটা
একটা নতুন লিংক জেনারেট করে করতে পারবে কি?
নতুন একটা লিংক জেনারেট করে
চাই না তো সবাইকে পাঠাতে হবে সঙ্গে সঙ্গে, না সবাইকে?
হ্যাঁ হ্যাঁ, এতজনকে পাঠানো তো একটুখানি এই মুহূর্তে।
কিন্তু যারা জয়েন করছে ওরা কিন্তু জয়েন করছে
না, ওরা স্যার আমাদের, এরা আমাদের মানে স্টুডেন্ট, এদেরকে জাস্ট ওই নেটে হোয়াটসঅ্যাপে
ডিরেক্ট লিংক দিয়ে জয়েন করানো হয়েছে আর কি, আর তো মানে সার্টিফিকেটের ব্যাপারটা আছে।
না, তুমি যদি যেভাবে আমাকে লিংকটা পাঠিয়েছো না হোয়াটসঅ্যাপে, ওই লিংকটা ওইভাবে যদি ওদেরকে লিংক পাঠিয়ে দাও, ও জয়েন করতে পারবে। আমি জয়েন করলাম ওইভাবে।
মোনালিসা, এটা সম্ভব এখন ওই লিংকটা দিয়ে দেওয়া?
হ্যাঁ হ্যাঁ,
হ্যাঁ অ্যাকচুয়ালি হ্যাঁ, লিংকটা আমি সেন্ড করে দিয়েছি। এবার মানে একদম লাস্ট মোমেন্টে তো লোকজন তারা হয়তো আস্তে আস্তে জয়েন করতে পারবে, এরকম একটা অবস্থা আর কি।
আমাদের স্যার লজ্জাই লাগছে আর কি বলতে।
হ্যাঁ, কিছুক্ষণ ওয়েট করো, কিছুক্ষণ আছে, 10 মিনিট ওয়েট করতেই পারো।
ঠিক আছে,
আচ্ছা
তাহলে আমরা 10 মিনিট।
আচ্ছা স্যার, হ্যাঁ
থ্যাংক ইউ। হ্যাঁ স্যার, সেলিম স্যার, আমরা 3:10 শুরু করি। হ্যাঁ, লিংকটা ডিরেক্ট শেয়ার করা গেছে। যারা জয়েন করছেন 3:10 আমি স্যারের সঙ্গে প্রফেসর জানার সঙ্গে কথা বললাম। 3:10 উনি তো মানে আমাদের সঙ্গেই আছেন, ওয়েট করছেন। আমরা 3:10 শুরু করি।
ঠিক আছে, ওকে ম্যাডাম।
হ্যাঁ। Namaskar sir. On behalf of Shabang Sajonikanta Mahavidyalaya, Department of Physiology, I am asking Dr. Shelem Chisti to inaugurate the session because our principal Dr. Tapun Dattu is in emergency. He is in the university, Vidyasagar University. So Dr. Chisti, our chairperson, IQAC cell of the college, Chisti will inaugurate the session, sir.
Thank you, madam. Good afternoon to all. Today we have assembled here on Google Meet platform on an international symposium organized by the Department of Physiology in collaboration with the IQAC cell of this Mahavidyalaya, Shabang Sajonikanta. Today, 4 distinguished research persons will deliver their lectures on augmentation of electrical and molecular milieu of human body with postural sequel, stratagem towards physiological crisis. We have with us Professor Nihar Ranjan Jana, IIT Kharagpur, Dr. Gautam Datta, PK College Kontai, Dr. Sandip Kumar Das, University of Gaurbanga, and Dr. Amor Kundu, Bombardier Transportation, United Kingdom. I welcome all these resource persons on behalf of our college. I also welcome all the participants of this international symposium. I ensure that today's symposium will be very fruitful to us. We, the participants, will be enriched today by their valuable speeches. Thank you very much. I am requesting the secretary of this international symposium to start this program. Thank you, madam. Hello.
Thank you, sir. We are already late due to some technical problem. I first welcome Professor Jana. He is a renowned person in his area. He is the first speaker of the session. I am feeling proud that I was also in the same college. He did his graduation from Midnapur College and post-graduation from Calcutta University. He did his doctoral studies at Vishwabharati University. After PhD, he completed his postdoctoral work in Riken Brain Science Institute, Japan. And then he joined National Brain Research Center as a faculty member. In 2001, he served as faculty at National Brain Research Center and then moved to IIT Kharagpur in the School of Biosciences. He has been awarded by Government of India a very prestigious award, National Bioscience Award for Career Development, one of the highest Indian Science Award in 2008, and National Academy of Science elected him as a fellow in the same year. He is also an elected fellow of the West Bengal Academy of Science and Technology in 2012. He received another prestigious award, that is Varsvik Industrial Research Award. So we are proud that he will spend some time, his valuable time with us. Welcome, sir.
Okay, so can I start? So thank you, Dhumu, for organizing this meeting.
Welcome,
long due for me. And good afternoon, all of you who have joined this meeting. I hope all of you can hear my voice. So I will just try to put my screen. So how long time? So we can. There are 4 speakers, right? So how long time you can? Can you hear me?
Yes, sir.
So there are issues. So I cannot.
Yes, sir. You are audible.
I am audible, but
Yes, sir, you are audible, sir. Sir, you can continue. Half an hour, 45 minutes. We are eager to hear you, sir.
Yeah, just I'm trying to share my slides, but I think something happening. It's not happening. I understand. Okay. So I hope you are able to see my slides, right? It's coming?
Yes.
Yes, sir. Yes, sir.
Oh, no. I think I'm still not able to. I think there are some issues. Let me try again. Okay, so I hope you are able to see my slides, right?
Yes, sir.
Yes, sir.
Okay, let it come. Yeah. So okay. So again, thank you all for joining this meeting. So I'll try to maybe half an hour to complete. So just to introduce, first of all, who are probably not familiar about IIT Kharagpur, particularly, the biology work that IIT Kharagpur is initiating in the last few years. So this is actually some of the part of the IIT Kharagpur probably is not the main building part. It's kind of outside the main building. So just to show you, this is where I'm putting my arrow. So that's the new actually building for biological science. This is called Life Science Building, or often called the Diamond Jubilee Building. And you can see the back side of there are a lot of construction going on where actually the medical institution is coming. And this year actually the first MBBS batch is joining, and class will start maybe from the next year beginning. So that's with that. So today I'm trying to kind of talk about a topic. I hope people will be interested to hear about that. It's on neural circuitry and neural plasticity. So when you think about our brain, just think and compare with the computer. So I'm just trying to give a brief overview. So you can consider our brain one of the most sophisticated computational machine, and it can change its own component, hardware component, which is the most amazing part. And itself can change in a new configuration whenever it does some new task. So there are our brain is about 1.2 or 0.3 kilogram weight that consists about 100 billions of neurons. And these 100 billions of neurons actually interconnected with each other to form a effective functional circuit that remains for life. Okay, and we have to remember that these circuits that are there in our brain, that's not static. It continuously changing. Whenever you are trying to learn something, when you are studying something or doing anything, like to remember something, our brain form completely new circuitry. So therefore, if you think that circuit is a hardware, it's not like a computer, it is static. It's continuously changing. And that's one of the phenomena, it's called plasticity. So my talk in the last part and second part, I'll explain how can we explain the plasticity in our brain and what its significance. So before that, I think I should first talk another part. People probably who are not very familiar, but I'll go a little fast because I'm sure many of you probably physiology students, so you have very good understanding about neuron. But I'll just quickly go through it: the discovery of neuron and visualization of neuronal circuitry. Today's whatever the advanced tools that are available, by using that tools, how can we understand, visualize the neuronal circuitry? So let me first introduce two very famous personalities and their work. And it's actually more than 120, 30 years ago. So one of the guy you can see here, Franz Nitschl, who actually discovered a dye. It's often called the Nitschl staining method. And the dye is nothing but some basic dyes. Sometimes you can see here about Kissel violet, thionine, like this. So they are the basic dyes. So they'll go and bind with the acidic component of the cell. So you can think nucleus and some nucleic acid that might be in the reticulum that's located. And by using this staining, you can actually understand various cellular morphology and cellular architecture. Like if you see this panel, this is a Nitschl staining brain part. It's actually part of the brain that actually store memories, hippocampus. And you can see the cellular architecture. Very simple, guys. The moment actually you can understand even cellular, morphology, right? But you can understand the time that he actually identified this and he has looked at this. That time neuron was not named, so you can think it's a cell of the brain. It looks like pretty other cell. There's no difference almost you can make out. But at the same time, there's another guy. It's called Camillo Golgi. We all know that his name. You can see the Golgi apparatus. But he also discovered another interesting dye or method. That's often called Golgi staining method. And in this case, what he did actually, he actually put a small piece of brain in silver chromate solution, maybe for a week or per month. And then if you cut a small slice, thin section, and watch under the microscope, you'll be able to see some of the cells in the brain. I'm not telling actually neuron, because neuron was not named that time. Some of the cells in the brain are dark structure, darkly stem cells, but that takes actually, huge branching. That's give you huge branching. And it was really amazing that time for him to see that some of the cells have extensive branching. Now you can compare these two staining method, and you can see what's the difference of the Golgi staining. By using Golgi staining, you can see the huge branching of a cell that it presents inside the brain. Okay. So that's actually one of the landmark you can discover, you can think in those era. It's more than 125, 30 years ago. Okay, so next, if you look at another guy, it's called Santiago Ramon Cajal. What he did actually, he used same Golgi staining method and looked at different parts of the brain and draw those images, whatever he saw in the brain, inside the brain. And in this case, he made the microscope that he used, actually his own handmade microscope. You understand? In those old era. So by observing those brain sections and drawing, this is what I'm showing the picture. This is actually his own hand drawing by observing under microscope. And you can see it's an amazing drawing. These are cerebral Purkinje neurons, and you can see how extensive branching they are. So what he found, he was confirming the observation of the Golgi that the cell in the brain have extensive branching. But other than that, he also noted some of the dot-like structure in the dendrite structure. So now the point comes. So when Golgi looked at the brain, by looking at his picture, he came to a conclusion. That's very important. The conclusion of Golgi was that two cells in the brain are connected with each other. It's a very continuous process, like a spider web. So information from one cell can go to the other cell directly. But Raymond Cahill, using the same staining method, come with a completely different conclusion. The conclusion of Raymond Cahill is that the cell from, the branching from or the one cell of the brain is not directly connected with another cell. There must be some gap. So you can now understand who is right. At that point, it was not really sure. Nobody was sure who was right. But both of them got Nobel Prize. Both of them got Nobel Prize, and it is actually one of the rare in the history of Nobel Prize that two persons got Nobel Prize with opposite and contradictory theory. Okay, so now you all know that Raymond Cahill was right. There's a gap, and gap is now you all know that's the synapse. So I'm not really describing that. I'll go fast on that. So you all know there's a gap, and that is gap is called synapse. That is actually given. That name is actually given by Charles Sherrington, followed by there's a discovery of chemical transmission, which is the story is actually unfolded by these two brilliant guys, Otto Louis and Henry Day. I'm sure many of you have already have studied that. But you have to also remember there is a excellent experiment done by Otto Louis. And the first time he demonstrated that there is a chemicals that is secreted from the vagus nerve that can slow down the heartbeat. And he actually don't know that time that it was what's the identity of the chemicals. He gave the name of that chemicals is vagus stop. This story you must read. So that's why you can get influenced. So the vagus stop later actually characterized by this Henry Dale, actually both of them, and now named actually acetylcholine. And acetylcholine is the first discovered neurotransmitter. Now you can see there are many such neurotransmitters actually discovered, and that is actually present in our brain. Okay, so this is just to give you a brief overview about that. Now, if you look at the structure of the neuron, that's there always. You can find out any class and textbook. I think you have probably seen in class 9, 10 textbook also. That's the typical cartoon picture. Two neurons joined, and there's a gap. That's called synapse. But now something that you have to know more details about it. Now, if you go much higher magnification, if you look at this dendritic area or axonic area, what you can see, and you can use the same Golgi staining. You have to remember this Golgi's method actually discovered say 125-30 years ago, and you can using that method and go much higher resolution, look more advanced microscopy. What you can see, I'm showing here in both left and right side, there are a lot of extended structure, extended protrusions. You can see that, and that is the structure is called dendritic spine. That's actually, you can see the arrow pointed out. So now you can think there's so many you can see. And if you can count, you will find out on an average, a single neuron might have thousands of such dendritic spines. Okay? And there are some neurons which might be having lakhs and 2 lakhs. Like if you look at cerebellar Purkinje neuron, where you might find out about 2 to 2.5 lakhs of dendritic spines. Now, the question is, what is the importance of these dendritic spines? So these are the structure, these are the extended structure where actually you will be able to see the formation of synapse. Therefore, dendritic spine indirectly tells you the number of synapses. So that if a neuron have about thousands of dendritic spine, you can think that neuron might be able to make about thousands of synaptic connections. Okay, so now you can visualize, you can think, and then you can think this cartoon picture, whatever you see in the textbook, is absolutely wrong. So now you can think a single neuron have about thousands of synapses, so it can make input, it can make connections about thousands different ways, right? So just try to visualize. Okay, so that's another very interesting point, and you can visualize this by just using a Golgi staining, and this is one of the method still I mean, the most advanced lab still using this. What I'm saying is, you can understand the power, what I will say, the powerful, it is so powerful. Somebody discovered this method 120 years ago. We still use this method and make a lot of new discoveries. Okay, so now if you look at much, I will say, detailed structure of this all this dendritic spine, you can see the bottom panel. This is actually the real image using Golgi staining method, and then you can just try to make a architect's drawing of that. And you see there could be different types of dendritic spines. There are thousands of dendritic spine in a neuron, and thousands of spine could be 1,000 different types. And from the types, you can make out which one is good, which one is bad, which one is mature, which one is immature. And I will not all the details, but if you look at the mushroom type, the mushroom type dendritic spine is the most mature one and does the most efficient synaptic transmission. There are other structures like filopodia, long thin, like this, they're actually very mature, and then these are processed, sometimes they are skin form, and they can be any moment eliminates. And now you think this dendritic spine, we all probably are very familiar about different kind of developmental disorder, and very commonly you can think about most autism spectrum, autism or autism spectrum disorder. In all those autism spectrum disorder, you can see there is a difference of this dendritic spine number. Different disorders has different, these autism spectrum disorders have different dynamics of this kind of dendritic spines, and that actually explains why a child that is suffering from this kind of autism spectrum disorder shows learning and memory impairment. So this is the bottom line. Okay, so now you can think about, right? So let me now. So this is actually very old techniques, right? But still, I mentioned, still people use repeatedly. And now I'll see what's going on. I'll show you what's going on in the field. So are there any other developed method that can have much better resolution to see the neural circuitry? So I'll just show you, there are a lot of development over the time going on, and I'll try to show you one of the method that's called Clarity, and that is developed primarily by this Carl Dissler from Stanford University. And by name you can understand, it actually clear the brain. So if you look at the brain, I mean, if you see, it's so opaque, highly dense, and it's almost 60 to 70% of our brain actually is full of lipid. So the idea is, can we make this brain transparent? And if you have to make the brain transparent, you have to actually remove all the lipids from the brain. So that's the idea. That's the idea people are working. Many people are actually on that. And so idea, what is the idea? So you have to remove almost all the fats from the brain, and at the same time you fix the hardware component. Hardware component is here the circuit, all the neurons, neural structures. So that has to be fixed and remove the fat. And then probably you can make your brain transparent if you want to make experimental. So that's how that guy did. That guy made a landmark paper that published in Nature 2013. So what they did, they used some kind of hydrogel which consists formaldehyde, which we commonly use to fix in tissues. You go in a museum, the tissues actually put in the formaldehyde. So formaldehyde, along with that, he used, it's called acrylamide. It's often used in making gel to run to separate proteins. So he used formaldehyde and the acrylamide monomer, make a hydrogel, and soak the brain for say 1 or 2, 3 days. And then you just increase the temperature to 37 degrees of that sample. So at that temperature, this gel actually, this whatever the monomer they use, is actually polymerized. And when it's polymerized, it actually cross-linked with many of the cellular protein. That cellular protein here, actually all the neuronal different kind of surface protein, it will go and cross-link. Thereby it makes the hardware, I'm talking about hardware always, because it's all the neuronal structure become intact, strong. Now the next step, you can actually remove the fat. How you can remove the fat? You can just put the brain in a detergent. If you put any, because it's a lipid, if you put this lipid in any detergent, the lipid will be solubilized and that you can wash out. If you put simply this brain in any lipid solution, in any detergent, you can keep 2-3 months the brain actually will be clear. But that takes so much of time, right? So what this guy did, this guy actually put this brain, this fixed brain, in SDS solution. So SDS is a negative charge. This is anionic detergent, anionic detergent. So they will go and bind with different lipid compounds. And now you put that in electric field. Now you put that SDS mixed brain sample in an electric field and pass the current. So what will happen? All the negatively charged SDS that is bound to the lipid will be moving towards the positively charged, right? Thereby can remove all the lipids from the brain, and thereby makes the brain completely transparent. And I will show you the next picture. Look at this. Hey, this is the picture where you see the brain has been put on a piece of paper. It is written something in English. I don't know whether you will be able to read, but some point you can read, right? Can't read. Now look at the B. There is actually brain sample. Same brain actually kept it here, but in this case, it has been after treated with clarity method. So you can see it is almost completely transparent. And now you can read the whole sentence. This is something is actually commented by Raymond Kahar. You can read it. The brain is, what is it in some world? Brain is a world consisting of number of unexplored continents and great stretches of unknown territory. This is the comment made by Raymond Carr. And now you can clearly read it. Okay? So, but you can't see anything, but you have to imagine that all the neuronal circuit, all the intact neurons are actually there. So now you can think about it in today's technology, transgenic and knockout technology, whatever available. So you can make all those neurons, each and every neuron in your brain with a different color. Like you can transgenically express in all those neurons the green fluorescence protein. What he did actually, he actually expressed transgenically YF, yellow fluorescence protein, in all the neurons. I'm not telling all the details, but then if you just pass UV light, you can see all those structures. Whatever you see completely clear, on the past UV light, you can see the fluorescence coming. And this on means on photon. So if you just pass through different intensity light, you can actually visualize a three-dimensional picture, image of brain. And if you go gradually higher magnification, you can see an excellent neural architecture. It's unbelievable. And if you see the end, you can actually see all the dendritic spine. Can you see all those dendritic spine? I think this picture may not give you real reflection, so I'll try to go to his lab. Hope I'll be able to show you, and just show you 1 or 2 minute clip. That can give you idea of what kind of neural architecture you can see. Look at it. This is his lab. I hope you are able to hear, right? The sound.
Only pictures, sir.
Hear, right?
Only we can visualize, but sound is not coming here.
You are not able to hear the sound. Oh. Okay, so I'll show. I'll just close after 1 minute. So I'll just probably see this has become clear. I'll just take you. Yeah, I think, and now you look at how you can visualize the three-dimensional way. I mean, neural architecture. Everything can be clearly visible. So even you can actually make different types of neuron in different colors, and then you can look at the circuitry. I mean, it's an excellent way to visualize almost entire brain, different kind of neuron, how they connected, everything you can see. So I'll just close this. So I'll go back to my slide again. Okay. So that's the first part. So now we are coming to the next part. That's the plasticity. So that I mentioned that our brain is continuously changing its hardware, and the phenomena that we can term it now is plasticity. If you look at the image here, this is actually image has been taken from a live animal, and the animal when is learning something, is the motor learning. You can see they are all dendritic spine. You can see at different time points, same neuronal, branching. You see there are two new spine appear when the rat actually trained to do some motor task. That tells you there are continuously new dendritic synapses are actually dendritic spines or new synapses are continuously formed. And just to give an example, now you are actually listening to the talk. At this moment in your brain, there are a lot of new dendritic spines are actually formed, and they're trying to make new connections. Okay, so you can understand the circuitry that is present in your brain is absolutely dynamic structure, and that's actually the plasticity. That's actually explain the plasticity. And you can think now, if there are 100 billions of neurons that I told you present in our brain, and each and every neuron might have about thousands of such dendritic spines or synapses. You can now think in our brain there could be total of 100 trillions of synapses. So now you can imagine how complex could be the circuitry. Okay, so that just you can think. So we're just trying to give you very basic aspect of the plasticity. So by now you understand what is the definition of plasticity. So it is the ability of our brain to change and adapt in response to environmental cues, experience, behavior, anything. And it is the mechanism by which our brain encodes and experience something new thing and learns a new behavior. It's not only that actually happens in normal circumstances, in normal individual, day to day. But you can think even this plasticity by you can actually correct, or by which a damaged brain can relearn a lost behavior when actually patient undergoes rehabilitation. Like think about a stroke victim who's actually survived but has been completely hand or legs paralyzed in the moment. But we can try to recorrect those problems by continuously putting rehabilitation. So the purpose of that rehabilitation is to make a new circuit in the brain. Okay. So now again I will try to explain this in different ways. This is very exciting. If you think seriously, look at. I'm just trying to show you some part of our brain. Think about the day you born. You have to go back. This is the part. This is the big part of the picture that tells you the day you born, and look at the number of neurons and some small branchings are there. Now there's another picture where you think you are probably 6 years old, and look at the number of. If you look at carefully, the number of neurons are fairly same. There is not much difference. But look at the difference. The extensive branching, right? And then again, look at the teenage when you become just cross teen. You see, of course, there are extensive branching. Number of neurons is almost constant, but the branching again came down. So now think about. If you try to kind of calculate and put a plot. Plot comes like this. So this is actually front side of the brain and the back side of the brain. The sharp peak, that's the back side of the brain, that's actually visual cortex. And front side, that's the frontal cortex, which actually stored off your higher cognitive function. I'm responsible for that. So now what do you think about? If you just compare all three different ages, you can think the number of neurons are fairly constant across the age. But what is the difference? The increased number of branchings, which indirectly suggests increased number of synapses at 6 years of age. That's why it's a peak, probably on an average. And then again it's come down. When you become teen, the number of synaptic connection again goes down, almost half. So if you look at this peak, if you look at this back side of our brain peak, it sharply goes up, maybe by 1 or 2 hours, years of our birth. Like if you look at our visual cortex, then again fall down by 8 years, it almost get half number of synapses. Then it just maintains. Pretty similar profile you can see in frontal lobe. So which tells you when you are probably in 6 years of age, that the time our brain actually extremely plastic, which means it's extremely sensitive to any kind of environmental stimulus. So that's what I wrote. So the plasticity in the brain changes with age. So early age, we will all never get back that the age all we have crossed was the extremely plastic. And that's the age you can learn anything about it. And the 6 years of age, that's the age actually is the most plastic our brain was. Okay, so now we'll try to explain what's the significance of that. Just look at now. If you just think about it, another terminology I will just bring the peak that I mentioned. Like if you look at the back side of our brain, the visual cortex, the maximum peak means I'm talking about maximum plasticity, or you can think indirectly the maximum number of synapses that actually happens about say 1 year after your birth, after our birth, or in the frontal cortex maybe about 8 or 9 years after birth. So these peaks are often called the critical period. I might have another 5 minutes, right? It's called critical period, right? So that's why you have to remember. This is in textbook. You will find out the critical period. So what is the critical period? So critical period now can be defined as the time window when our brain is extremely sensitive to environmental influence. Okay, and now you can think 6 years of age. You can't remember. You can ask your mom. What are the things? What are the notorious things you did actually? You'll be amazed to see probably hear that. Go and ask your mother. What are the things you did during your, that's the time actually probably you first try to work, try to learn a new language, and anything what probably you thought you can do that time. And it's actually possible that time. So I'll try to give you the history of that, how this concept of critical period came. I think it's a very amazing story for all of you probably. Let's look at all those bars, cheeks, turkeys, goslings, whatever. The interesting phenomena probably have not noticed. You must have looked at all these pictures, but whatever probably have not come across your mind. Whenever they hatch, whenever the egg they come out after hatching of the egg, within probably 2-3 hours of hatching, these goslings actually start walking and just follow their mother. That's the phenomenon probably I have never watched. But one of the interesting things this scientist did is actually ornithologist Conan Lawrence. What he did actually, instead of their mother, of this gosling's mother, he just stand there and start walking. What he noticed that goslings followed him, and he repeatedly do this and perform this experiment and come to a conclusion that within 2 and 3 hours after hatching of the goslings, something must have happened in their brain that allow them to follow any moving object. It's not their mother only. It can be any moving object. If you stand in front of them and walk, they will follow you. So that's a landmark. I mean, it's a very simple story. You can see in any rural part, rural Bengal. I mean, this incident probably very difficult to nowadays see. But in our time, we used to often see this. But from the simple observation, you can see from the landmark discovery actually came out. But the important thing you have to remember that within 2-3 hours after hatching, something must have happened to their brain that allow them to walk any moving object. And this phenomena will not be taking place maybe after 5-6 hours. So only small window, only few hours. After that, it never happens. Okay, so that's how the critical period idea came. But if you look at the mammals, particularly monkeys, and for us, this problem didn't happen. This actually, this imprinting phenomenon didn't happen. But very similar thing happens, and if you just think and some of the example you can understand. So you can think we are always fond of our mother, right? Not really the father. Mother will be very happy to hear. And that's one of the reasons you can think about that from the day of your birth, you are continuously with your mother. It's not only the visual stimulus. I think visual stimulus actually make secondary. I think sometimes the smell and the feeding baby, those are some of the incidents that makes the child much closer to their mother. And there are a lot of increasing evidence. I mean, there are increasing number of papers nowadays appearing that says even the milk consists the prolactin. When mother feeds the baby gets prolactin, and when the baby gets prolactin actually has huge impact in making the new neural circuit, and thereby making an attachment with mother. Social bond it actually makes in the early stage. And you can again, there are series of experiments people have done. Mother who didn't feed their baby, their surrogate mother, in that case probably childs are not that making bond. So there are a lot of experiments have been done in monkeys. Okay, I hope that's clear to you, but this might be very, very interesting aspect. Okay, so there was time, so I will not really discuss about that. But now if you look at overall scenario of our brain, so most of the sensory structures like vision, hearing, all those sensory development actually come first in the critical period. Then actually your different other part of the brain like higher cognitive functions or emotional functions, their critical period actually comes much later. Okay, so probably I will not have time, but you can now experimentally prove. You can experimentally prove how this plasticity can take place in the brain, and that two guys, that Preston Wiesel and David Hubel actually proved that by using visual cortex of kitten. So in case of kitten, I'll just briefly talk about the in case of kitten. The critical period about something mentioned about 2 to 4 weeks probably. So during that their immature time and their critical time, if you close the eyes, just twist the eyes of the kitten, you can see their visual cortex is permanently changed. So you can close and open the eyes and you can do a lot of experiments. That's what the experiments are there, they have done. So you close one eyes and then for 2 days, and then you do the experiment and you will be able to see the secondary in the visual cortex at the backside of your brain is actually irreversibly changed. Okay, so I'll not really, this is the experiment that did actually. So I'm mostly going to be done, right? So I'll just probably put more focus on what's advantage. What's the advantage? Because this is one of the hot area of brain science research, the critical period. So purpose is, we know that by 6 years of our age, the peak actually start falling down. So the idea is, what are the things you can learn in this? And because the brains are extremely plastic during the critical period, you can do actually a lot of things. So the advantage, so that's why I wrote here, critical period can be a window of opportunity as well as vulnerability. So during this early phase, you can actually make your child, if you try to teach your child, you can make your child smarter and smarter. There are some of the examples I gave you. The earlier days, we used to grow up in a joint family. So there are just so many people in the family. Forget about if mother don't take care, there are a lot of other people are there. So you need to continuously expose with them. That's one of the excellent environment that often called the enriched environment. And nowadays, it's very hard to think. Both father and mother goes to different jobs, and child could be in a daycare center or with somebody who's taking care of the babies, and sometimes may not be taking care properly. That makes a child much more vulnerable. So that's the vulnerable stage. So the window can be advantage. You can teach good thing during the time child can learn. On the other hand, a bad thing can go also in that time. If any bad thing goes during that, this critical period window, the child can be even bad child. So that's one of the, I think, important message for even just think about in our country, most of the child, from the pregnancy to birth and to the developmental stage, so much of malnutrition. Okay, it's not the only undernutrition. Sometimes the rich people also have malnutrition because of the overnutrition. That also kind of malnutrition. So both of the situation gives huge problem in our country. Okay, so this is the bad advantage and disadvantage. The best advantage, I think I can give the example is the learning a language. Learning a language, right? You can think about if you try to learn a new language at this age. If you now try to learn another language now, I don't think it's practically impossible for any of us. But your child can do. Child which is about 2 to 10 years of age can learn any language. So it's a good idea for any mother or father, any parent, if you want to teach your parent multiple languages, to teach together. When they learn the local language, their mother tongue, at the same time, you could teach as many as language, they will quickly adapt. And that's how probably many daycares, if you look at all those nursery schools, try to do it. At least 3 languages is easy to learn. So I think that's the biggest advantage of critical period. So I'll probably try to stop with this, and I hope this one will work. This is probably 1 or 2 minute one video that probably you can get it from NIS side in Harvard Medical School. Who are interested can just watch. Again, probably you will not able to hear the sound, right? Probably just look at. So what I'm trying to explain is that the joint family, how they can huge impact in the brain. How mother and child communication can extensively change the neural architecture in child's brain. It's unfortunate that you are not able to hear the sound, otherwise it would have wonderful. Just look at, there's a new circuit is formed. So there's a new circuit, strong synapse. You see synapse are lost here. Okay, so I think you can probably look at in Harvard Medical School website. I think it's already there. So I'll stop. If you go to this place, you can get this kind of YouTube videos and you can look at it. So I am probably done with time. So Juma, I'm done. So people might have questions. I don't know whether they can ask or not. Hello. Yeah.
Thank you, sir.
Yeah. If you have questions, can definitely ask.
Thank you, sir. To the audience, if you have any question, you feel free to share with Professor Jana. Sir,
Yes.
I have a question. I
Sure.
want to speak in Bengali because so many
yeah,
students
yeah.
are there.
Yes.
Actually, sir,
Right.
it is plasticity or spastic. And
Yes.
I want to experience some expertise view on these two terms. Sir.
Plasticity and plastic. So you can after almost similar pattern on same time. So plastic মুড়ি দিচ্ছ মুচকুচে যাচ্ছে দেন ছেড়ে দাও আবার নরমাল চলে আসছে মানে এটা হচ্ছে যে তুমি তোমার ব্রেনটাকে যা খুশি করতে তুমি মুচড়ে যা করতে চাও করতে পারো
আবার প্রিভিয়াস ফর্মে ফিরে যেতে
পারবে এন্ড সেটা সম্ভব কখন সবচেয়ে বেশি হতে পারে যখন তোমার মানে ছোট ছেলেদের যখনই তুমি টিন এজে পৌঁছে গেছো টিন এজের পর লাইক অ্যাডাল্ট এজ যখন হয়ে গেছো তখন ওই মানে চেঞ্জ করার ক্ষমতাটা ব্রেনের কমে যাচ্ছে আস্তে আস্তে যখন 6 বছর বয়স বাচ্চা সেইটাই হচ্ছে বেস্ট টাইম মানে ম্যাক্সিমাম চেঞ্জ ক্যান তখন ওই সময় হতে পারে
আরেকটা স্যার এই যে স্প্যাস্টিক যেটা সেক্ষেত্রে নিউরাল যে সার্কিট গুলো নতুন নতুন তৈরি হচ্ছে লার্নিং এক্সপিরিয়েন্স করছে বিভিন্ন জিনিস তখন নতুন সার্কিট হচ্ছে স্প্যাস্টিক যাদেরকে বলি আমরা তাদের ক্ষেত্রে কি এই সার্কিট ফর্মেশনটা কোথাও হ্যাম্পার্ড হয়
তুমি স্প্যাস্টিক বলছো রাইট স্প্যাস্টিক স্প্যাস্টিক সেক্ষেত্রে কম হতেই পারে ইভেন তুমি যদি দেখো যে কোনো ওই চাইল্ড যারা জন্মেছে ওই ডিফেক্টিভ চাইল্ড মানে ডিফেক্ট যাদের ওই ডাউন সিনড্রোম হতে পারে বা অটিস্টিক চাইল্ড তাদের ক্ষেত্রে তো প্রচন্ড ভাবে এফেক্ট করে আমি বলেছিলাম যে সার্কিট গুলো অ্যাবনর্মালি ফর্ম মানে সাইনাপস গুলো অনেক কমে যায় এই অনেক সময় অনেক জায়গায় কমে অনেক জায়গায় বাড়ে যেমন তুমি যদি অটিজম চাইল্ডে দেখো অটিজম চাইল্ডের ক্ষেত্রে দেখো ওদের ক্ষেত্রে কিছু কিছু পার্ট ব্রেন প্রচন্ড বেড়ে যায় সেই জন্য তুমি অনেক জায়গায় দেখবে অনেক অনেক চাইল্ড অটিস্টিক চাইল্ড অ্যাকচুয়ালি এক্সট্রা অর্ডিনারি মানে ব্রিলিয়েন্ট
হাইপারও হয় এরা অনেক সময় মানে ভীষণ মুভিং হাইপার চাইল্ড
এত হাইপার মেইনটেইন করাটা মানে কন্ট্রোল করাটা কখনো কোনো ভীষণ ডিফিকাল্ট হাইপার
এটা স্যার আমি দেখেছি কারণ আমাকেই স্যার ট্রেনে একবার একটি এরকম অটিস্টিক বাচ্চা চুল ধরে প্রচুর মেরেছিল উইদাউট এনি প্রভোকেশন প্রচুর পিটিয়ে দিয়েছিল আমাকে থামানো যায় না ওদের আরেকটা জিনিস স্যার এটা একেবারেই আমি জানি না একদম মানে খুব চাইল্ডিশ প্রশ্ন হলেও করছি সেটা হচ্ছে স্যার আমরা যখন এই লিঙ্গুইস্টিক ডিভিশনটা নিয়ে একটু একটু ঘাটাঘাটি করি তখন বলছি যে ডিফারেন্ট ল্যাঙ্গুয়েজ শেখার সময় তাদের সার্কিট গুলো আলাদা আলাদা ফর্ম করে
সেটা ঠিকই কিন্তু অলমোস্ট সেম জায়গাতে ল্যাঙ্গুয়েজ এরিয়া একটা ব্রেনের মধ্যে দুটো তিনটে জায়গা আছে মোস্টলি দুটো জায়গা যেখানে ল্যাঙ্গুয়েজের রিলেটেড ইনফরমেশন গুলো ওখানে জমা হয় তো যখন তুমি ধরো বাংলা শিখছো আমি জাস্ট এক্সাম্পল দিচ্ছি বাংলা আর হিন্দি তুমি যদি একসঙ্গে শিখো সিমিলার টাইপ অফ ল্যাঙ্গুয়েজ হয় বাংলা আর হিন্দির মধ্যে সিমিলারিটি আছে যদি ল্যাঙ্গুয়েজ একসঙ্গে শেখাতে পারো কোনো বাচ্চাকে they are actually going the same place same brain area তে অ্যাকচুয়ালি ওখানে গিয়ে নতুন সার্কিট তৈরি করছে মানে এটা হচ্ছে অনেক ইজি
হ্যাঁ, একটা বয়সে ইজি হচ্ছে। একটা স্যার বলছে যে কিছু ভাষা আমরা বলি কঠিন, কিছু ভাষা সহজ। এই যে ফিলিংসটা একটা টু লার্ন ইজি, টু লার্ন ভেরি ডিফিকাল্ট, এই যে ডিফারেন্সটা হচ্ছে, এটাও কি ওই সার্কিট ফরমেশনের সঙ্গে কোনোভাবে রিলেটেড হতে পারে?
খুব একটা নয়, এই ডিফারেন্সটা আমরা বলছি যখন আমরা অ্যাডাল্ট হয়ে গেছি, মানে আমরা যখন পারছি না বুঝতে। তুমি যে একটা বাচ্চাকে বলো, তুমি যদি আজ যদি চলে যাও সে নর্থ সাউথের ল্যাঙ্গুয়েজ আছে, অন্ধ্রপ্রন্ধ ল্যাঙ্গুয়েজ, তেলেগু বললে সামটাইমস আমরা ভাবি সো ডিফিকাল্ট, কি করবো কি লেখা আছে। কিন্তু যদি একটা বাচ্চা, একটা বাঙালি ছেলে ফ্যামিলি ওখানে জন্মায়, ওখানে ছোটবেলা থেকে তৈরি হয়, ও ইজিলি অ্যাডাপ্ট করে নেবে, ওর কাছে ওটা কোনো কমপ্লিকেটেড মনেই হবে। আমি আমার বাড়ি এক্সাম্পল দিয়ে বলছি, আমি আমার মেয়ে জন্মেছে, আমি যখন 2 বছর দিয়েছি, আমার মেয়ে ওখানে জন্মেছে, তো ও বাংলা জানে না, তো ওকে আমরা বাংলা হিন্দি জেনারেলি প্রথম থেকে স্কুলে যাচ্ছে, সবার সঙ্গে মিশেছে হিন্দিতে, তো হিন্দি ওর কাছে ইজি। বাট আমি হিন্দি আমি বলতে পারি, বাট আই ক্যান স্পিক, আই ক্যান রিড, সরি আই ক্যান রাইট, বাট ও বাংলা ইংরেজি যেহেতু বাংলাটা আমার মা ওর কাছে শিখছে, তো বাংলা হিন্দি ইজিলি অ্যাডাপ্ট করেছে, হিন্দি ওর কাছে কোনো প্রবলেম হয় না। সেরকম আমি কেউ যদি সাউথে যদি জন্মায় বাচ্চা, যখন তৈরি হচ্ছে ও ওখানে সবার সঙ্গে মিলেমিশে সবার সঙ্গে করছে, ওর পক্ষে ওটা খুবই ডিফিকাল্ট নয়, আমাদের কাছে মনে হচ্ছে খুবই ডিফিকাল্ট। যে কোনো ল্যাঙ্গুয়েজ ওদেরকে পড়ে দেওয়া ও শিখে নেবে, দ্যাটস দ্য এজ,
মানে ওই এজটাই অ্যাকচুয়ালি এজটাই ফ্যাক্টর,
এজটাই ফ্যাক্টর। 10-12 সাল পেরিয়ে গেলে আর পারবে না, 10-12 সাল পেরিয়ে গেলে পারবে না, কেন আমাদের কি বলে এখন পারবো না, কিন্তু ভীষণ ডিফিকাল্ট, এখন অন্যরা বুঝতে পারছি। মানে এটা যারা পার্টিকুলারলি যারা নিউ মাদার, যারা তাদের পক্ষে ভীষণ ইম্পর্টেন্ট to make the child to learn all the language, as many as language, শুধু ল্যাঙ্গুয়েজ নয়, even চাইল্ডের বিহেভিয়ার, and this very এখনকার সমাজে I think it is খুবই ইম্পর্টেন্ট। মানে how much time আমরা আমাদের ছেলেদের দিই, আমরা দিতে পারি না, মানে সবাই চাকরিতে চলে গেছে, ছেলের সঙ্গে কোনো ইন্টারাকশনই নেই, এই রাতে এসে কোনো সময় নেই, তারপর তুমি অফিসে কাজে ব্যস্ত হয়ে বিরক্তিকর। So how you deal with the child, that's very important. চাইল্ড যখন বড় হবে ও কি কোন ব্যবহার করবে, it's very important.
অ্যাকচুয়ালি স্যার মানে কম বয়সটাই হচ্ছে তাকে শেখানোর পক্ষে বা কিছু অ্যাডপ্ট করার পক্ষে উপযুক্ত, এটা খারাপও হতে পারে ভালোও হতে পারে।
আমাদের সময় আমরা স্কুলে যা করেছি, এখন তো স্কুলে আমি জানি না সেই ধরনের হয় কিনা, বাট আমাদের সময় তো স্কুলেই যা শেখা শিখেছি আমরা।
না এখন স্যার বাড়িতেই যা শেখা শিখে, আর টিভি আছে, তারপরে মোবাইল আছে, ওখান থেকে শিখে নেয় সব।
মোবাইল ইজ গুড অলসো ব্যাড অলসো।
হ্যাঁ স্যার, আমাদের তো একটা এক্সপেরিয়েন্স হয়েছে গতকাল স্যার, আমি শেয়ার করছি এই কারণে যে মানে জাস্ট এই প্রসঙ্গটা এলো বলে, আমাদের স্টুডেন্টরাও আছে এই প্ল্যাটফর্মে। আমরা গতকাল আমাদের ভ্যাকসিনেশন ক্যাম্প চলছিল লাস্ট ডে, তো সেখানে হোয়েন উই আর আস্কিং কোন স্ট্রিম, আমরা দুটো আলাদা আলাদা করে নিলে আমাদের ভেরিফিকেশনের সময় সুবিধে হয়। তো এখন যে যারা ফার্স্ট ইয়ারে ভর্তি হলো, একদম কলেজ শব্দটাই যাদের কাছে নতুন ইন্ট্রোডিউস হচ্ছে, তারা কেউ বলতে পারছে না তারা কোথায় অ্যাডমিটেড হয়েছে, বিএ ক্লাসে অর বিএসসি ক্লাসে অর বিকম ক্লাসে, তাদের স্ট্রিমটা কি তারা বলতে পারছে না। আমাদের চেক করতে হচ্ছে, মানে ওরা পেন আনে না, ওরা এত বেশি অনলাইন অ্যাডাপ্টেড হয়ে গেছে যে ওরা পেন ক্যারি করছে না আর কি সঙ্গে। এটা অদ্ভুত এক্সপিরিয়েন্স হয়েছে, একজন ভর্তি হয়েছে কিন্তু সে বিএ বিএসসি বা বিকম বলতে পারছে না, আমাদেরকে কাগজ সার্চ করে দেখে নিতে হচ্ছে। এটা শেয়ার করলাম স্যার।
কমন, ভেরি কমন। সো প্রবলেম হচ্ছে মাদার পেরেন্টস উইল হ্যাভ টু ফেস দ্য কনসিকোয়েন্স, আফটার সামটাইমস পেরেন্টসদের প্রবলেম ফেস করতে হচ্ছে। বাট এই ছেলেরা ইভেন যখন তারা নিজেদের লাইফে প্রবেশ করবে, এখন তো বাবা মার আন্ডারে, সো যখন নিজেদের লাইফে যাবে, অ্যাডাপ্ট হবে, মানে they will face huge problem in the life, day-to-day life.
আর স্যার একটা লাস্ট মানে কোয়ারি আর কি, কোশ্চেন না স্যার, একদমই কোয়ারি। ইজি ব্যাপারটা যেটা আমরা বলি ডিউরিং অ্যাকশন, কোনো একটা নির্দিষ্ট কাজের সময় কেউ কিছু একটা শিখছে, কিছু পড়ছে বা কিছু করছে নতুন কিছু, সেই সময় ইজি প্যাটার্ন যেটা চেঞ্জ হয়, এটা কতটা রিলায়েবল, মানে এই যে ডেটাটা আমরা পাবো এটা কতটা আমাদের কাছে গ্রহণযোগ্য?
ভেরি ভেরি রিলায়েবল, মানে যদি ঠিকমতো ইজি নেওয়া হয়। To encephalogram, so the people should know that whenever you are trying to measure electrical activity in the brain, এটাকে বলা হয় EEG। হ্যাঁ, তুমি যদি ঠিকমতো ইলেকট্রোড গুলো প্লেস করো, ব্যাকগ্রাউন্ডটা ভীষণ ইম্পর্টেন্ট, কিন্তু অনেক সময় ব্যাকগ্রাউন্ড ওয়েভটাই এমন করে দেবে যে তুমি আসল ওয়েভটা বুঝতে পারো। তো ঠিকমতো যদি ইলেকট্রোড গুলো প্লেস করা হয়, আর ঠিক মতো যদি ব্যাকগ্রাউন্ডটাকে কারেক্ট করো, ইজি অনেক ইনফরমেশন দেবে। মানে তুমি নর্মালি যখন অ্যাক্টিভিটি করছো, জানি ভালো করে, যখন আমরা ঘুমাই বা যখন আমরা জাগি, ইজ প্যাটার্ন ডিফারেন্ট। মানে ঘুমানোর সময় যখন স্বপ্ন দেখে বা ডিপ স্লিপ বা রেম স্লিপ বা নন রেম স্লিপ এগুলো হয়, সেখানে তো ইজি প্যাটার্ন প্রচন্ড ডিফারেন্ট হয়ে যায়। তুমি যখন দিনে ডিফারেন্ট কাজ করছো, ইজি প্যাটার্ন ভ্যারিজ। এখন অ্যাকচুয়ালি ইজি প্যাটার্ন দেখে অনেক সময় অনেক কিছু ডিজিজ ডায়াগনোসিস করা হয়।
যেটা করতে পারফেকশন উইথ পারফেকশন কাজটা করলে পাওয়া যেতে পারে। এখন অনেক মডার্ন টেকনিক্স এসে গেছে, তো সেই জন্য
এখন মনে করি তুমি বুঝতে পারবে যে তোমার ব্যাকগ্রাউন্ড কোথা থেকে আসছে বা অনেক বুঝতে পারবে।
ইজি রিলায়েবল বলা যেতে পারে।
ভেরি মাচ লাইক ইসিজি, মানে ইসিজি তো ঠিকমতো যদি প্রপারলি কেউ রেকর্ড করতে না পারে, ইসিজি কেন যে নটোরিয়াসলি মানে প্রবলেম আছে। মানে তুমি একটা পারফেক্ট পারসন, একজন ইসিজি করে বলে দেবে তুমি একজন তোমার প্রচন্ড হার্টের প্রবলেম। আমি তোমাকে বলছি তোমার আমার ওয়াইফ তোমার বন্ধু, রাইট? ওর একটা সার্জারি জায়গায় ইটস জাস্ট নরমাল ইসিজি করতে হবে। এন্ড ইসিজি করে ডক্টর বললো ইউর হার্ট ইজ অলমোস্ট ফেল, মানে চাঁদা অবস্থা। সো ফরগেট অ্যাবাউট সার্জারি, হার্ট নিয়ে বাঁচতে। উই গো অন টু নেক্সট ডক্টর, another places to do ECG is absolutely fine. So it depends কি করে তুমি রেকর্ড করছো। সো তখন বুঝলাম যে নন স্পেসিফিক্স কি করে হতে পারে।
হ্যাঁ,
এটা ভেরি কমন। মানে আমাদের বেঙ্গলে এটা ভেরি কমন না। সুতরাং তুমি যেটা কোশ্চেন করলে, ইসিজি ইজ ভেরি রিলায়েবল।
থ্যাংক ইউ স্যার। আর কারোর কোনো প্রশ্ন আছে? একটা প্রশ্ন পেয়েছি স্যার। ব্রেন প্লাস্টিসিটি, এজ এবং মেমোরি, এদের মধ্যে কোনো মানে কংক্রিট রিলেশন আছে কিনা একটা একজন জিজ্ঞেস করছে।
প্রথম কথা হচ্ছে, আমি দেখলাম কোশ্চেনটা, ব্রেন প্লাস্টিসিটির সঙ্গে এজ তো রিলেশন আছে। যত তুমি বড় হবে, যত তুমি বয়স বাড়বে, টিনেজটা হচ্ছে একটা টার্নিং পয়েন্ট। টিনেজের পরে মোটামুটি একটা স্টেপ। আদারওয়াইজ মানে বয়স যখন তোমার খুব ছোট, তোমার প্লাস্টিসিটি খুবই ব্রেন প্লাস্টিসিটি খুবই আছে, এটা বললাম। বয়স বাড়লে আস্তে আস্তে কমতে থাকবে। ইভেন তোমাদের এজ, যারা মানে স্টুডেন্ট যারা, ওদের এজ আর আমাদের এজ কম্পেয়ার করি, ওদের ব্রেন অনেক বেশি প্লাস্টিক, ওরা অনেক বেশি আগে অ্যাডাপ্ট করতে পারবে। একটা জিনিস যদি পড়ি আমরা, ওরা অনেক বেশি মেমোরাইজ করতে পারে আমাদের থেকে, আমাদের সময় লাগে। সুতরাং এজের সঙ্গে তো ব্রেনের প্লাস্টিসিটি ভীষণ রিলেশনশিপ। আর একটা মেমোরি, ইয়েস অবভিয়াসলি। আমি চাইল্ড, তুমি যখন চাইল্ড যে লিখেছে, তুমি যখন চাইল্ড ছিলে, আমি বললাম তো মাকে জিজ্ঞেস করো তুমি কি নটোরিয়াস অ্যাক্টিভিটি করেছো, যেটা তুমি এখন করতে পারো। ওই সময় তুমি যা শিখতে পারবে কম সময়ের মধ্যে। যখন আমরা স্কুলে যখন পড়ি, ওই যে টেবিল নামটা যখন পড়তাম, এখন পড়ি না জানি না, আমরা ক্লাস লাস্ট পিরিয়ড থাকতো আমাদের ওই নামটা মুখস্থ করা, মনে আছে? ওইটা আমরা কোথায় বই পড়েছি, আমরা ওইখানেও প্র্যাকটিস হয়ে যেত। সুতরাং চাইল্ডহুড টাইম তোমার ব্রেন ইউ আর এক্সট্রিমলি ব্রিলিয়েন্ট। তুমি কারোর সঙ্গে কারো কম্পেয়ার করতে পারবে না যে ওর ছেলে ভালো, আমার ছেলে খারাপ। ওই স্টেজে হয় না, এটা হয় একমাত্র হাউ ইউ মেক ইউর চাইল্ড। একটা সার্টেন স্টেজের পরে এগুলো হয়। অফকোর্স জিন হ্যাজ এ ব্রেন। I mean, কোন জিনিয়াসের ছেলে জিনিয়াস হবে, you don't think. And look at our prime minister, he is not from a genius family. So there are hundreds of examples. I mean, if you look at in Midnapur district, there are people who are doing extraordinary work. Very good people are there. Then you look at, they are from very poor family.
And first generation learner. In most of them are first generation learner.
গরিব বাড়ি থেকে ধান কেটে খেয়ে বড় হয়েছে, দেখো তারা কোথায় থাকে।
একদম।
সুতরাং অফকোর্স যে কোশ্চেন বলছে, চাইল্ড ডেফিনেটলি প্লাস্টিসিটির সঙ্গে মেমোরি ইজ ভেরি মাচ ডাইরেক্টলি প্রপোর্শনাল।
থ্যাংক ইউ স্যার। আমার মনে হয় এটা আমার স্টুডেন্টদের কাছে, আমাদের সবার কাছে একটা মেমোরেবল ডে আজকে স্যার এক্সপিরিয়েন্স আপনার সঙ্গে কাটানো অনেকটা সময়। আজকের প্ল্যাটফর্মটা একটু তো স্যার ঝামেলা হয়েছে, কারণ আমাদের এই টেকনিক্যাল প্রবলেমের জন্য। তো
অনলাইনে হচ্ছে,
আমাদের রুরাল বেল্টের ছেলেমেয়েরা একটু ভালো কিছু এক্সপোজার পায় স্যার আপনারা এলে। হোপ স্যার আপনি পরেও আবার আসবেন আমার সাথে। হ্যাঁ স্যার, তেমাথালি
আমার মামা বাড়ি, সো আই নো দিস কলেজ ভেরি ওয়েল।
আমরা যখন অফলাইন হব স্যার, একদিন এলে খুব ভালো লাগবে আমাদের সবার।
I think
Thank
অন্যদের
you,
টক
sir.
আছে, ওদেরকে দিতে হবে টক না, অন্যদের লেকচার
sir.
আছে না।
ঠিক আছে স্যার, থ্যাংক ইউ।
ঠিক আছে, তো আমি ক্লোজ করছি আমার।
ওকে। Our next speaker, Dr. Gautam Datta. Hello.
Am I audible?
Yes, sir. Yes, Gautam, you are audible.
Okay.
Welcome, Gautam. Dr. Gautam Datta, Assistant Professor, Department of Physiology, Prabhat Kumar College, Kontai, Purbo Medinipur. We have heard him in 2019 in our college. He is a good speaker. I can recall his throwing, the quality and the pattern of throwing, and I am his fan, no doubt, fan of his topic and the presentation, way of presentation, to say better. So he is awarded Young Scientist Award from Physiological Society of India in 2009. He has 2 books. He wrote the 2 books, 16 peer-reviewed journal publication. The title of his talk today is Sleep-Wake Pattern During COVID-19 Pandemic Lockdown. আমার স্টুডেন্টদের জন্য আমি একটুখানি বলছি যে এই যে প্যান্ডেমিকের সময় তো আমাদের, স্যার ভুল হলে একটু কারেকশন করে দেবেন, যে প্যান্ডেমিকের সময় আমাদের সবারই ডেইলি রুটিন একটুখানি চেঞ্জ হয়ে গেছে, আমরা একটুখানি অন্যরকম রুটিনে অভ্যস্ত হয়ে গেছি। তো সেই বোধহয় স্যার এইটা সম্পর্কেই বলছেন যে এই সময় যে ঘুমানো জেগে ওঠার প্যাটার্নটা কিভাবে এফেক্টেড হয়েছে, প্রবাবলি স্যার সেই সম্পর্কেই বলবেন। আমি আমাদের স্টুডেন্টদের রিকোয়েস্ট করব এটা শুনতে খুব মন দিয়ে, এবং স্যার ভীষণ ভালো বলেন, আমরা আগেও শুনেছি স্যারেরটাও ভীষণ ভালো বলেন। ওয়েলকাম স্যার, ইউ ক্যান কন্টিনিউ।
Thank you, the Organizing Secretary of this international webinar, Chuma Madam. I am also thankful to the Honorable Principal of Sajinikanta Mahavidyalaya, commonly known as Shabam College or Temathan College, and all the organizing team of this webinar. And thank you, Nihan sir, for your very informative and very important talk. As a neurophysiology student, this is very much important and new for me till now. Thank you, Sandeep, the University of Telangana, Sandeep Kumar Das, and I also thankful to Amit Kundu who are present and my speaker, for this webinar. And not the last, not the least, my dear students. And so as you are all not the physiology student, I think, so I think my lecture will be better if you deliver it by language. The Bangla এবং ইংলিশ মিলিয়ে বলি তাহলে একটু সুবিধা হয় বুঝতে, কারণ ল্যাঙ্গুয়েজটা যদি প্রবলেম হয়ে যায় তাহলে আর শেখার কিছুই থাকে না। স্যারও বললেন যে ল্যাঙ্গুয়েজ হয়তো আমরা সেই সময় ভালো করে শিখিনি 6 বছর বয়সে, তখন যদি আমাদেরকে একটু ইংলিশ এটা শেখানো হতো ভালো করে, হয়তো আমরা আরেকটু ভালো বলতে পারতাম। এগুলো পরে আমরা সবই অ্যাডাপ্ট করেছি ইংলিশ বলাটা অ্যাজ ইন্টারন্যাশনাল ল্যাঙ্গুয়েজ। তাই এখন যারা স্টুডেন্ট আছো তারাও চেষ্টা করো। এখনো স্যার বললেন যে প্লাস্টিসিটি এখনো আমাদের মধ্যে আছে, তোমাদের মধ্যেও আছে, তোমাদের স্টুডেন্টের এজে। তো অ্যাডাপ্ট আর প্লাস্টিসিটি দুটোকেই কাজে লাগিয়ে এখনো শেখা যায়, তাই শেখার চেষ্টা করা উচিত সবসময়। তো আমার টপিকসটা আজকে যেটা রিলেটেড, দ্যাট দ্য স্লিপ ওয়েক প্যাটার্ন ডিউরিং কোভিড-19 প্যান্ডেমিক লকডাউন। তো লকডাউনটা শুরু হয়েছিল সম্ভবত মার্চ 2020। আমরা আজকে অনেকটা পথ চলে এলাম। এখানে অনেকগুলো ব্যাপার প্রথমে একটু বলে রাখি, যে আচ্ছা ফার্স্ট স্লাইডটা দেখিয়ে বলি যে একটা ডিফারেন্স আমরা কিন্তু দেখতে পাচ্ছি ছবিটাতেই, যে একটা স্লিপ, যেটা কার্টুন পিকচার, যে স্লিপ আমাদের কিন্তু প্রতিদিনের একটা রুটিন, এটা ডেইলি লাইফ। স্লিপ ওয়েক সাইকেল আমরা মেনটেন করি, চলি আমাদের দিনে। আর পাশের দুটো ছবি কিন্তু একদম আজকের দিনের মডার্নাইজেশনের একটা ফল। এখন লেট নাইট ওয়ার্কিং এটাও একটা ট্রেন্ড, কারণ খুব বাস্তব বলতে গেলে বাড়িতে থাকলে সারাদিনের পারিবারিক কাজে হোক বা অন্য কাজে হোক অনেকটা সময় চলে যাওয়ার পর আমার মনে হয় রাত্রি করে কাজটা করে নেব। And the main culprit is the social media. I think so. The Facebook, Instagram, WhatsApp, all are the main culprit behind this alteration of sleep cycle during this COVID-19 situation. The dependency on online mode is the problem. উপায় নেই কিছু করার, তাই আমরা এটাই এসেছি, সুইচ অফ করেছি, এখানে সুইচ অন করেছি আমরা অনলাইন মোডে। কিন্তু এর ভালো এবং খারাপ দিকগুলো দেখতে গেলে এর খারাপ দিক একটা বড্ড আমার চোখে পড়েছিল। অ্যাকর্ডিং টু আমার স্টুডেন্টদের উপরেই আমি কিছু জিজ্ঞেস করেছিলাম। তো সেটা করতে গিয়ে আমি যেটা দেখলাম যে অনেকে রাত্রে বা সকালের ক্লাসগুলো করে না। আমার কিছু ক্লাস মর্নিং, আমাদের কলেজে মর্নিং ক্লাস হয় বলে আমি কিছু মর্নিং ক্লাস থাকে আমাদের 8টা থেকে, তো অনেক স্টুডেন্ট মর্নিং ক্লাসটা করে না। তো একদিন আমি খুব ফ্র্যাঙ্কলি ওদের জিজ্ঞেস করেছিলাম, ওরা বলছে আমরা কেউ ঘুম থেকে উঠতে পারি না। তো হোয়াট ইজ দ্য প্রবলেম? তো বেশ কিছু স্টুডেন্টের একটু ভালো যারা মিশতে পারে তারা বলেই দেয় স্যার অনেক রাত্রি অব্দি জেগেছিলাম তাই উঠতে পারি। তো জেগে থাকার কারণগুলো যখন ওদের সঙ্গে আলোচনা করে পাওয়া যায় তখন বেশিরভাগের কারণ কিন্তু ওয়াচিং বা ইনভলভমেন্ট ইন সোশ্যাল মিডিয়া, ফেসবুকের বিভিন্ন পোস্টিং, ইনস্টাগ্রাম আর গুলো বললাম না, সেই সবগুলো মিলিয়ে আজকে একটা কিন্তু বড্ড বাজে জায়গা তৈরি হয়ে গেছে। তো সেই জায়গাটায় তো কালপ্রিট কি এই জিনিসটাই, তো এই কারণে প্রেজেন্টেশনটা এরকম করলাম যে করোনা কি তাহলে কোনোভাবে আমাদের এই স্লিপ ওয়েক প্যাটার্ন চেঞ্জের কালপ্রিট, এবং তার ফলে আমরা কি অন্য কোনো ফিজিওলজিক্যাল প্রবলেমে ভুগছি? এটা একটা বড় প্রশ্ন আমার মনে এখন হয়েছিল। তো আমি যদি একটু পেছন দিক থেকে আসি, যে ঘুম এবং জাগা এই দুটোর যদি আমরা একটা তফাৎ করতে চাই, যে আমরা খুব সহজেই জানি আমরা কখন ঘুমোই কখন জানি, রাত্রে ঘুমোই, তাহলে হোয়াট ইজ দ্য ডেফিনেশন অফ নাইট? আই থিঙ্ক দ্য ডার্কনেস ইজ দ্য ডেফিনেশন অফ নাইট। কারণ এই ছবিটা দেখে যতটা রাত্রি মনে হচ্ছে, আমি যদি এই ছবিটা দেখাই এটা একটা রাত্রির ছবি, কিন্তু then what is the basic difference between two pictures of night? This is the darkness of night, and this is the lightness or the brightness of the night. কথাগুলো শুনতে অন্যরকম লাগলে আমার মনে হয় যদি কখনো আমার ঘুম পায় তাহলে কিন্তু I choose this picture, কারণ this is the real symbol of night. তাহলে নাইটটা যদি আমার ডার্কনেস হয়, তাহলে এই পিকচারটা যদি আমার ডার্কনেসের যদি অপোজিট হয়, তো আমরা একটু তাহলে একদম এনসিয়েন্ট টাইমে ফিরে যাই, যে এই ঘুমানো জাগার এই প্যাটার্ন এগুলো কিন্তু মডার্ন লাইফের নয়, এগুলো কিন্তু ফিজিওলজি। ফিজিওলজি তো আজকে আমরা মানুষ নয়, ফিজিওলজি অনেক পেছনের দিক থেকে মানুষের ফিজিওলজি অ্যাডাপ্ট হয়ে 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 আজকে আমরা এসেছি। তো তখনও কিন্তু দিন রাত্রি তো হতো, এটা তো আর নতুন করে বলার কিছু নেই।
স্যার, গৌতম স্যার, অ্যাকচুয়ালি কোনো সাউন্ড আসছে না।
হ্যাঁ, গৌতমটা মানে লেফট করে গেছে, কিছু একটা প্রবলেম হচ্ছে মনে হয়।
হ্যাঁ, নেটওয়ার্কের প্রবলেম আছে আজকে।
হ্যাঁ,
একটু
জয়েন করে
ওয়েট
যাবে।
করি আমরা হ্যাঁ
হ্যাঁ।
একটু ওয়েট করি।
কিছু লো ফ্রিকোয়েন্সি, সেইভাবে আমরা ভাগ করে দিই, যেমন বেটা ওয়েভ, আলফা ওয়েভ, ডেল্টা ওয়েভ। তো অ্যাওয়েক এবং রেম স্লিপ এই দুটো কিন্তু অনেকটা সিমিলার, এবং রেম স্লিপটাকে আমরা ড্রিম স্লিপ বলি। এই নিয়ে অনেক আজকে রিসার্চ হয়, কেন হয়, কোন কে রেসপন্সিবল, এখানেও নিউরাল প্লাস্টিসিটি কিভাবে রেসপন্সিবল, এই নিয়ে অনেক কাজকর্ম হয় এখন নিউরোফিজিওলজিতে।
গৌতমদা,
তো এখানে হ্যাঁ
গৌতমদা, হ্যাঁ বলছি তোমার শেয়ারিংটা বন্ধ হয়ে গেছে। হ্যাঁ, তোমার শেয়ারিংটা একটু বন্ধ হয়ে গেছে, মাঝখানে একটু অফ হয়ে গেছিল। তুমি একটু শেয়ার করে দাও আরেকবার।
আছে, কোথায় আছে?
প্রেজেন্টেশনটা আসছে না স্যার।
এখন দেখা যাচ্ছে?
হ্যাঁ, এবারে দেখা যাচ্ছে। হ্যালো, হ্যাঁ হ্যাঁ ইউ আর ভিজিবল স্যার।
ওকে, তো এই জায়গাটা যেটা ছিলাম যে বিভিন্ন স্লিপ টেস্ট গুলো নিয়ে একটু বলছিলাম, যেহেতু স্লিপ ওয়েভ সাইকেল তাই একটুখানি বলে নেওয়া দরকার ছিল। তো এই স্লাইডটা কিন্তু ইম্পর্টেন্ট, আমরা একটা কিন্তু সার্কাডিয়ান রিদিম দেখছি, দ্যাট ইজ দ্য ফ্রম মিডনাইট টু নুন। এই পুরো 24 আওয়ার সাইকেলকে আমরা যদি ভাগ করে দিই, তাহলে কিন্তু এখানে স্লিপ ওয়েক এবং ডিফারেন্ট অ্যাক্টিভিটিস আর গোয়িং অন। তো এর মধ্যে একটা জিনিস ইম্পর্টেন্ট, দ্যাট ইজ মেলাটোনিন। কেন? এটা তো আমরা ফিজিওলজিতে একটু জানি যে মেলাটোনিন এমন একটা হরমোন যেটা রাত্রেবেলা সিক্রেশন হচ্ছে, যেটাকে আমরা হ্যালোইন হরমোনও বলি অনেক সময়। There is a secreted in the night, a darkness hormone, আর এটা বন্ধ হচ্ছে যখন আমরা জেগে যাচ্ছি। তাহলে এখানে একটা রিলেশন ভালো করে মনে রাখা দরকার, যে অন্ধকার হলে ঘুম মেলাটোনিন হরমোন সেখানে আসছে, আলো আসবে মেলাটোনিন হরমোন বন্ধ হচ্ছে, সেখানে কিন্তু ছেড়ে যাওয়ার কথা। তো এই ফ্রম মেলাটোনিন সিক্রেশন থেকে শুরু হওয়া থেকে বন্ধ হওয়া, এই জায়গাটাকে কিন্তু আমাদের মেনটেন করা উচিত, যেটা জেনারেলি স্লিপ প্যাটার্ন হিসেবে আমরা জানি। তো যদি আমরা সেটা মেনটেন না করি, তাহলে স্লিপ ডিপ্রাইভেশন বলে একটা কথা আছে, যেটাও আজকের দিনে খুব ইম্পর্টেন্ট, যে বিভিন্ন কারণে আমাদের স্লিপ ডিপ্রাইভেশন হয়। আমরা ঠিক সময় ঘুমোতে পারি না, লেট নাইট ঘুমোই, বেলা করে উঠি, বিভিন্ন স্ট্রেস এন্ড ডিপ্রেশন, ডিপ স্লিপটাকে অ্যাভয়েড করায়। ওই জন্য যে স্লিপের কোন কোন জায়গাগুলো ইম্পর্টেন্ট, ডিপ স্লিপ ইজ ভেরি ইম্পর্টেন্ট। আমরা যদি স্লিপ স্টেজ উঠতে চাই, তাহলে কিন্তু আমাদের ঘুমের যে ইম্পর্টেন্স যে ইম্পর্টেন্স অফ স্লিপ ইন ফিজিওলজি সেগুলো আমরা কিন্তু পাবো না। যে মাসেল রিপেয়ার হচ্ছে কি অন্যান্য মেটাবলিজম স্লো হচ্ছে, তাই বা হরমোনাল যে রেগুলেশন যেটা স্লিপের সঙ্গে রেগুলেটেড কোনোটাই কিন্তু হচ্ছে না। তো সেটা যদি হয় তাহলে কিন্তু ফিজিওলজিতে বিভিন্ন ধরনের কিন্তু অ্যাবনর্মালিটি আসতে পারে। সেটা নার্ভাস সিস্টেম থেকে শুরু করে ডাইজেশন থেকে শুরু করে বিভিন্ন সব জায়গায় যদি আমরা রিসার্চ নিয়ে পড়াশোনা করি, ফিজিওলজিক্যাল একটা কিন্তু বড় চেঞ্জ আসে এবং হরমোনাল মধ্যে কিন্তু একটা আমরা that is the best background. ব্যাকগ্রাউন্ড একটু জাস্ট বলছিলাম স্লিপ ওয়েক কেন ইম্পর্টেন্ট, কারণ এটা ফিজিওলজির সঙ্গে রিলেটেড। এই স্লিপ ওয়েকটা কিন্তু একটা সার্কেডিয়ান রিদম মেইনটেইন করে চলে, যেটা নাইট এন্ড ডার্ক এবং আলোয় অন্ধকারের সাথে রিলেটেড। কিন্তু এবারে সবার আমরা আদৌ সেটা মেইনটেইন করছি কিনা। এবং এই মডার্ন আজকের দিনে লকডাউন সিচুয়েশনে I think the coronavirus is hiding the clock. The clock is hiding by the coronavirus. আমরা কিন্তু ঘড়িটা প্রায় ভুলতে বসে গেছি। কেন ভুলতে বসেছি বলছি, ভেরি বেসিক এক্সাম্পল আমার ডেইলি লাইফ দিয়ে, আপনাদের সবার লাইফ দিয়ে ভেবে দেখলে বুঝতে পারবো যে হ্যাঁ, হাতে 24 ঘন্টা সময়। যখন অফলাইন ছিলাম তখন একটা টাইম বাউন্ডিংস ছিল, এখন সেই টাইম বাউন্ডিংস কিছু নেই। যখন যা ইচ্ছে করছি রাত্রেবেলা জেগে করতে পারি। তো ঘড়িটা কিন্তু হারিয়ে গেছে, যেটা ইজ হাইডিং বাই দ্য করোনা ভাইরাস। এবং তার জন্য যেগুলো এলো সেগুলো লেট নাইট ওয়ার্কিং, তার জন্য ডিপ্রেশন আসছে, ভালো লাগছে না করতে, বাউন্ড টু ডু দ্যাট, প্রেশার ফ্রম দ্য অথরিটি। জেগে আছি, রাত্রি জেগে সকালে ওঠার তাড়া নেই, কলেজ যাওয়া নেই, ওয়ার্কিং স্পেসে যাওয়া নেই, রাত্রি জেগে টিভি দেখছি এন্ড সোশ্যাল মিডিয়া। এটা তো বারে বারে বলছি, এটা কিন্তু প্রত্যেকের কাছে আজকে একটা কিন্তু কালপ্রিট। তো এই সবগুলো থেকে আমার যেটা মনে হয়, ইন মাই পয়েন্ট অফ ভিউ, উই আর গোইং টু বি নকটার্নাল। আমরা কিন্তু নকটার্নাল অ্যানিমেলের দিকে যাচ্ছি, কিন্তু আমাদের ফিজিওলজিটা কিন্তু এই ধরনের নয়। দিস ইজ দ্য বেসিক ডিফারেন্স। The nocturnal animals physiology and their hormonal control, their night-day cycle is totally different from us. কিন্তু আমরা যদি রাত্রিগুলোকে না ঘুমিয়ে যদি এই ধরনের আলোতে এক্সপোজ হতে থাকি দিনের পর দিন, এবং বিশেষ করে দ্য মোবাইল, দ্য স্ক্রিন যেটা খুব চোখের সামনে থাকছে, তার ফল কি হতে পারে। এখন ফিজিওলজিতে ফল কি হতে পারে সেটা খুব একটা বড় ব্যাপার, খুব রিসার্চের ব্যাপার। তাই আমি এখানে অন্যরকম ভাবে প্রেজেন্ট করলাম। ইন দ্য বিগিনিং অফ দুর্গাপুজো আমরা দুর্গা মাকে যদি স্মরণ করতে পারি, তাহলে একটা জায়গা মনে করতে পারি আমাদেরও একটা থার্ড আইস আছে। এটা ফিজিওলজিতে আমরা পড়েছি কলেজে, প্রফেসর জানাও মেদিনীপুর কলেজের স্টুডেন্ট, ওখানেও আমরা পড়েছিলাম। তখন আমার একটা কথা মনে আছে এখনো, তাই আমি এটা বলি, দিস ইজ দ্য পিনিয়াল গ্ল্যান্ড, ইজ দ্য থার্ড আই। তো এই আমাদের দুটো চোখ দিয়ে আমরা দেখতে পাচ্ছি আলো, সবই দেখতে পাচ্ছি, কিন্তু এই আলো আর অন্ধকারের আসা যাওয়াটাকে কিন্তু সেন্স করে একটা অন্য চোখ, দ্যাট ইজ দ্য পিনিয়াল গ্ল্যান্ড, যেটা মেলাটোনিন সিক্রেশন করে। ওই মেলাটোনিনটাই এমন একটা হরমোন যেটা আমাদেরকে ঘুমে এবং অ্যাওয়েকেনের মধ্যে একটা ব্যালেন্স করে দিচ্ছে। তো এটা না হওয়ার জন্য কিন্তু অনেকগুলো কারণ হতে পারে, যে আমরা যদি এই সাইকেলটা মেনটেন না করতে পারি আমাদের বেসিক ফিজিওলজিটা হ্যাম্পার হচ্ছে। কিন্তু সব বেসিক ফিজিওলজির ব্যাপারগুলো বলা সম্ভব নয়, ভেবে ওঠাও সম্ভব নয়। তাই আমি একটা হরমোনাল ইমব্যালেন্সকে কিন্তু হাইলাইট করেছিলাম, এবং সেই ব্যাপারটা নিয়ে আমি যখন একটুখানি ভাবি, তো দেখলাম যে মেনস্ট্রুয়াল সাইকেলের উপর কোনো এফেক্ট আছে কিনা। তো এই ব্যাপারে আই এম ফরচুনেট দ্যাট ইন 2020 I was selected as a mentor of CSIR online research training program. তো তার সেই দৌলতে কিন্তু আমি অনেক স্টুডেন্ট পেয়েছিলাম, 28 জন স্টুডেন্ট পেয়েছিলাম ফ্রম ভেরিয়াস পয়েন্ট অফ ইন্ডিয়া। সেখানে কেরালা স্টুডেন্টও ছিল, আবার কাশ্মীরের স্টুডেন্টও ছিল। ভেরি ইন্টারেস্টিং ওয়াজ দ্যাট। তো সেই স্টুডেন্টদেরকে যখন আমি একটু নিউরোফিজিওলজি ইইজি বা তার টেকনিকগুলো বলছিলাম, তখন আমার মনে হলো যে তারাও এই ব্যাপারটা নিয়ে ভুগছে, যে রাত্রেবেলা সকালে ক্লাসগুলো দেরি করে করুন স্যার। তো আমি বললাম, তো ভাই নট ডান অনলাইন সার্ভে। তো একটা অনলাইন সার্ভে করলাম যে এই ঘুমোনোটা কিরকম করছে ওরা, বিশেষ করে কলেজ স্টুডেন্টরা, আর তাদের মেনস্ট্রুয়াল সাইকেল আর অন্যান্য কিছু ব্যাপারে কি চেঞ্জ হচ্ছে, একটা অনলাইন সার্ভে করি। এবং অল ইন্ডিয়া বেসিসে আমি 931টা স্যাম্পল পেয়েছিলাম। তো সেটাকে অ্যানালিসিস করে যেটা পেলাম সেটাই আমি একটু বলি আজকে তাহলে। যে এখানে এইটা প্রেজেন্টেশন করেছি তার কারণ যে মেনস্ট্রুয়াল সাইকেলটাকে আমি কেন ধরেছিলাম, কারণ এমনিতেই বেসিক্যালি আজকের দিনে এটা একটা প্রবলেম ডিউ টু স্ট্রেস হোক, ডিপ্রেশন হোক, ফুড হ্যাবিট হোক, ডেইলি লাইফের হোক, যাই হোক এটা একটা নাওয়াডেজ মেয়েদের কাছে খুব একটা বড় প্রবলেম হয়ে গেছে। তো বেসিক কিছু রিসার্চে গেলাম না, জাস্ট আমি রেজাল্টটা দেখাই। এখানে রেজাল্ট একটু ভালো করে দেখো, যে স্লিপ ডিউরেশন দেখেছিলাম এবং কখন ঘুমোতে যাচ্ছি। তো বিফোর কোভিড এই ব্লুটা হচ্ছে 7 ঘন্টার বেশি ঘুমানো, লালটা হচ্ছে 7 ঘন্টার কম ঘুমোচ্ছে কারা। তো বিফোর অ্যান্ড আফটার কোভিড ডিফারেন্স করে দেখা গেল যে করোনার প্যান্ডেমিক লকডাউনে এই আফটার কোভিড মানে এটা কিন্তু আমি করেছি 2021 এর জানুয়ারি। তো এই সময়ের এক্সপিরিয়েন্স থেকে যেটা পাওয়া গেল যে বেশি সংখ্যক কিন্তু কলেজ স্টুডেন্টরা, বিশেষ করে ফিমেল স্টুডেন্টদের উপরই এটা করা, তারা কিন্তু বেশিরভাগ 7 ঘন্টার বেশি ঘুমোচ্ছে। সো হোয়াট, ঘুমোতেই পারে, দ্যাট ইজ নট দ্য প্রবলেম। বাট বেসিক প্রবলেম ইজ দ্যাট তারা কখন ঘুমোতে যাচ্ছে আফটার 12। আফটার 12 মানে কিন্তু 12টা 1 মিনিটও হতে পারে, দ্যাট ইজ দ্য নট ম্যাটার। কিন্তু আমি দেখেছি এখানে 1টা, 1:30টা, 2টো এরকম গুলোকেও আমি 12টার পরেই ধরেছিলাম। তো যেটা ছিল 26% বিফোর কোভিড, 12টার পরে ঘুমোতে যেত 26%, সেটা আজকে 60%। 60% স্টুডেন্ট আজকে 12টার পর ঘুমোতে যাচ্ছে। সেটা 12টার পর মানে বলছি আমি, একটা আছে, দুটো আছে, দুটোর পরেও আছে। তা সেখান থেকে যদি 7 ঘন্টার বেশি ঘুমোয় তাহলে কিন্তু তারা উঠছে প্রায় 8:30, 9টা বা আফটার 9। তাহলে আমাদের যে পিএমএন সাইকেলটা ছিল না, দ্যাট ইজ ট্রান্সফার টু এএমএন সাইকেল, and the melatonin hormone secretion is totally hampered. And তার জন্য আমি irregularities of menstrual cycle দেখেছিলাম। Irregularities means আমি দেখেছিলাম যে less than 25 days and more than 35 to 36 days cycle। তো সেখানে আগেও ছিল irregularities, থাকবে না তা নয়, but the irregularities is increased 25% after COVID or during COVID lockdown. So are there relations between these altered sleep behavior with this menstrual cycle? তো সেটা দিয়ে আমি কি অ্যানালিসিস করে দেখলাম, এবং there is a published in Indian Journal of Community Medicine that yes, there are relations. Those who are going to sleep after 12 or after 1 and 2 and their waking time is after 8 or 9 a.m., they are more prone for these irregularities. তারা কিন্তু irregularities ভুগছে। আর তার মধ্যে আর দুটো প্যারামিটারও আমি দেখলাম, দ্যাট ইজ দ্য ডিপ্রেশন। ডিপ্রেশন বলছে 46.35%, এটা কোভিড এর আগে কিন্তু অনেকটা কম ছিল। ভ্যালুটা আমার এক্স্যাক্টলি মনে না থাকলেও সেটা 20 ঘরে ছিল। ডিপ্রেশন তো থাকবেই স্টুডেন্টদের মধ্যে, কিন্তু সেই ডিপ্রেশনটা বেড়ে গেছে 46.35%। তো এর কারণ হতে পারে বাইরে না বেরোতে পারা, একটু বদ্ধ হয়ে যাওয়া, হতে পারে বাড়ি বা আমাদের সঙ্গে বেশিরভাগ সময় কাটানো খারাপ নয়, কিন্তু তাদের মনের ইচ্ছেটা পূরণ হচ্ছে না। স্ক্রিন টাইম ইজ, হোয়াট ইজ দ্য স্ক্রিন টাইম? 78.5% স্টুডেন্ট বলছে তাদের স্ক্রিন টাইম বেড়ে গেছে, এবং সেই স্ক্রিন টাইমের পরিমাণটা কিন্তু বেশিরভাগটাই রাত্রে। কারণ এখানে বলতে কোনো লজ্জা নেই। melatonin may be exogenous or endogenous, that there may have some potential effect on this damage of screen quality or sebaceous gland problem or hair quality. So at the end of this slide, I described the COVID as a new form that is circular rhythm of our sleep physiology. It's very effective on irregularities of menstrual cycle and female reproductive physiology. My dear student, তোমাদের একটাই request, please maintain the sleep work cycle. There may have some effect on your menstrual cycle, which may be affect your reproductive life and further, or the polycystic ovarian syndrome. Ovarian syndrome nowadays is a burning problem to all of you. So you are very requested to please maintain the sleep work cycle as much as possible, and don't use your mobile phone in front of your eyes during the night time, because this light may hamper your melatonin secretion, which may alter your reproductive life in upcoming future. এত বড় কথাটা বলাও যদিও কোনো সাইন্টিফিক বেসিস আছে কিনা জানি না, কিন্তু I think in physiology the prevention is better than cure. The prevention is better than cure. So be safe and be careful. একদম শেষ স্লাইডে what to do or what to not to do, বাকিটা not to do আমি যেটা 12টাকে লাল করে দেখালাম, don't do like that. The corona is not going as totally, so please don't use your mask like this, and যদি ঘুমাতে হয় এভাবে ঘুমাও, and exercise and mask wearing is very important nowadays also. So thank you to all of you, and over to you, madam.
থ্যাংক ইউ স্যার। আমি আগেই আমার স্টুডেন্টদের বলেছিলাম, আমাদের ডিপার্টমেন্টের স্টুডেন্টদের বা যারা আছেন, যে অ্যাকচুয়ালি আপনার বিষয় এবং সেটাকে প্রেজেন্ট করার যে পদ্ধতি, এটা আমাদের সবার ভীষণই ভালো লাগে। আরেকবার আমরা সেটা পেলাম আপনার কাছ থেকে। তো আমাদের যদি স্টুডেন্টদের কারো প্রশ্ন থাকে বা কারোর কোনো প্রশ্ন থাকে, একটু স্যারের সঙ্গে শেয়ার করুন। স্যার, এত সহজ করে বলবার জন্য বিষয়টা আমার মনে হয় যে স্টুডেন্টরা খুব ভালো বুঝেছেন। Actually they don't have any question. So again, thank you sir. Thank you to be with us. In future we hope we may join together offline. Okay. Thank you sir. So this is the term of our next speaker, Dr. Sandip Kumar Das. Welcome, Dr. Das. Actually, Dr. Das is the student of our university, Mother University, Vidyasagar University. He obtained his doctoral degree from the Department of Human Physiology with Community Health. His specialization is in immunology and microbiology. He had 57 research articles in the field of nanotechnology, cancer biology, immunology, and bacteriology. He has developed several nano formulations for selective targeting of different cancer cells, as well as clinically isolated multi-drug resistant bacteria. His current research area emphasizes the modification of plant-derived tetrabonads for making it highly selective for cancer cells and cancer stem cells. He is also involved for fabrications of various metal and organic nanoparticles using green synthesis, and to investigate their efficacy against drug-resistant superbugs. Actually, very interesting area, I think. Along with lab-based experimental works, he is also performing survey works based on socio-demographic factors relating with COVID-19 pandemic. Dr. Das has several distinguished positions in the university in academic and administrative organizations. He has 3 registered PhD scholars. He is also acts as an executive editorial member of Health Outlook Journal, Journal of Cancer Clinical Research, and Journal of Experimental Medicine and Biology, published from USA, and reviewer of various reputed journals. So I am requesting Dr. Das to deliver his talk before us. It is your turn, Dr. Das. Welcome.
Thank you, madam, for such a nice introduction. I want to express my sincere thanks to the entire organizing committee of Sabong Sajanikanta Mahavidyalaya and IQAC department for organizing such a wonderful international webinar. I also congratulate my senior Dada Gautam Datta. Dr. Datta is also associated with our department. He is one of the visiting faculty, and every year he tried to and he engaged in the teaching of first-year students, especially in the nervous system topics. And I also found my some of my classmates here also present, and some of my pass out students and running current batch students also there in the online platform. And one of my friend also faculty in this college, Dr. Mona Lisa Banerjee. So thank you all. So I am just opening my presentation. Is it visible? Is it visible?
Yes.
Yes, sir. Yes, sir. It is visible, sir.
Okay. Thank you. So my presentation is basically based on one of the major question which has been found after the initiation of vaccination programs throughout the country. This question is based on the types of the vaccine which we want to select among the Covaxin and Covishield. Covaxin and Covishield, these are the 2 main vaccines which was introduced in India to combat the deadliest virus, which is known as the SARS-CoV-2, and in the short form COVID-19. So in this slide, this is my university of Gaurbanga. I am associated with this university in the Department of Physiology. I have joined here in the 2016. And from then, there was since the joining, there was the 2 small rooms were there, and after day by day with continuous effort, we have successfully established 1 cell culture unit, 1 microbiology unit, 1 molecular biology unit in our within a very short span of short space in the department. So it is a very beautiful department, and the beautiful atmosphere also there. So I came to my main topic. I will not take too much time. Just I will try to give a brief idea about the which vaccine is more superior among the Covishield and Covaxin. So at first, the coronavirus, that is the SARS-CoV-2, was first identified in the Wuhan, one of the city of China, in December 2019. The cluster of the pneumonic cases came out one after another, and latterly it was termed the COVID-19. In considering the infectivity pattern, considering the infectivity rate, in the 30th January 2020, World Health Organization declared is the health emergency of international concern. And on 11th March 2020, the World Health Organization again it is classified as a pandemic based on the infectivity pattern, the death rate, and other important parameters. So what is coronavirus? So we already know that it is a crown-shaped spike protein present on the surface of the virus. There are several types of crown-shaped spike proteins. That's why it is called the coronavirus. This virus having the particle size about 29.9 kilobase per, and it is basically a RNA virus, consists of a very large genome, uh, and the main structural protein consists of the spike protein, the membrane glycoprotein, envelope proteins, and hemagglutinin, elastoprotein, nucleic acid photoprotein, etc. So, so why we need vaccine? Because the vaccine is the only way to generate the specific antibody against this virus. Before vaccination, several types of drug like remdesivir and various types of commonly used drug like hydroxychloroquine and combination of the several antiretroviral drugs were applied to combat the disease. But initially, even though they showed some degree of success rate, but this was not consistent. So the main focuses of the research are placed towards formation of more appropriate and more potency containing vaccine. So uh, it's a very good news for us that within a very short period of time, a number of the vaccine has already been approved for combat against those disease. And the India is mainly dealing with two types of vaccine, that is Covishield and Covaxin, and recently one vaccine, that is Sputnik 5 vaccine, recently introduced in some parts of the India. It is also still in the still not given in the very large scale. And why vaccine is necessary? So vaccine is types of the adaptive immunity. So you will find that there are numerous blogging, numerous advertisement, numerous forwarding messaging, numerous advertisement from the company also, and that they want to share their products like immunoboosting food, immunoboosting tonic. But basically, for boosting of the immunocompetent cells, which are mainly responsible for the production of the antibody, is entirely depends on the antigen challenges. So these immunoboosting foods or immune enhancing substances basically are the types of antioxidant foods. They does not improve or does not generate the specific antibody. They only can suppress the oxidative stress, and thereby the metabolic status of the immune competent cells of the body are function normally, and they can perform the effective protection, but they do not have direct action towards the generation of the adaptive immune response which vaccine does. So vaccination is the only way to prevent such deadliest virus. So COVID-19 vaccination update, this data has been taken from the government website, Ministry of Health and Family Welfare, and and from this data, it is very good to share that among the 6 crore population in the West Bengal have been already vaccinated. And among them, 4.5 crores given the dose 1, and dose 2, nearly 2 crores have received the dose 2. So it is a very good data for us because a majority of the population has become vaccinated, and it is also good to share that the major section of the people who are not even vaccinated, and recent studies suggest that they have already become immune to this virus. This because from the herd immunity. So there is a general question among the people. Basically, I found in various website news or various bloggings and various communication from also the various communication, and also when I discuss about the vaccination to my colleagues, to my students, they sometimes ask me, "Sir, which vaccine is best?" And I found there is still no study where it has been critically analyzed and proven that which vaccine is best among the Covishield and Covaxin. Why I don't discussing other vaccines, because the in India Covishield and Covaxins are the major two types of vaccine which has been administered widely. So, to know the awareness levels, the acceptance levels of Covishield, Covaxin, and other types of vaccines in the specifically the common people in the West Bengal, we have conducted a survey in 22 February to 22 March 2021, and there are nearly 800 individuals participate in the survey, where the Google form link has been circulated and distributed using the WhatsApp group, using the social platforms, and also using the personal contacts. Among 803 individual, 435, which is nearly 54.17% male, and 368, which is nearly 45.83% female participants. They participate in this survey, and the result is very interesting. They give their vote in the favor of the Covaxin. Nearly 60% of the people they want to take the Covaxin, or and whereas only 26.90% of people they express their desire to take that they want to take the Covishield. Among the various age groups, that is less than 25 years, 25 to 39 years, and greater than 40 years, in each and every cases, a higher percentage of the people prefer the Covaxin, which is one of the indigenous vaccines, and other percentage of vaccine like the Pfizer, Sputnik 5, it is very small, within 1 to 2% only. So, to discuss the efficacy rate of the two vaccines, we should study the immune response against COVID-19. What is the general immune response of our body, which shows the protective mechanisms against COVID-19? So we know the coronavirus or SARS-CoV-2 virus mainly entered through the cells using ACE2 receptor, and using this ACE2 receptor, this enter into the cells, and thereby they stimulate the innate immune response and adaptive immune response. This picture has been drawn by my students, and the entire study has been communicated in the journal Immunological Research. It is in under review status. Here you can see the SARS-CoV virus engulfed by the lung epithelial cells, which is also a ACE2 receptor positive cells, and after entering to these cells, it triggers the viral particles also uptaken by the various antigen presenting cells. By the professional antigen presenting cells, we know there are three types of professional antigen presenting cells present in our body, that is the macrophage, dendritic cell, and B lymphocytes. And lung epithelial cells also can express the MSA class 1 molecules. And firstly, I want to discuss the innate immune response. The innate immunity refers to the immune response which we get from the very early stage of our life, just after the birth. And this immunity is mediated by the various surface innate immune receptor like PAMs, that is the pattern recognition receptors. These receptors generally activate the macrophages, and these macrophages become activated, and they release various types of chemokines, interleukins, like TNF alpha, interleukin 6, interleukin 8. This causes further activation of the macrophages, and which also can potentiate the activation of the dendritic cells. The activated macrophage can easily digest the virus using phagocytosis. The DC also takes the virus like same ways, and the generated interleukin beta can also activate the neutrophils, and neutrophils releases several types of antiviral proteins like perforins, enzymes, interferon gamma, and this also tries to kill the virus which has been entered through in our cells. So, on the other hand, when we talk about the adaptive immune response is mainly performing by the MSC class 2 positive cells. That that means the entire virus which has been entered into lung epithelial cells become digested by the antigen presenting cells, and the epitopic part has been represented by the MSC class 2 molecules, and TCR recognize them, and then the population of the TCR become converted to the cytotoxic T lymphocyte, and another population of the T helper cell converted to the Th2 types, and which are are responsible for the generation of the various types of immunological response. The cytotoxic T lymphocyte, we know that cytotoxic T lymphocyte are very important immune competent cells which gives the protection against the viral infection. And on the other hand, the humoral immunity, which also potentiate to using the MSC class 2 molecules, and this generate the plasma cells, which are the continuous antibody producing cells and the memory B cells. And this the viral information stored in the memory B cells, and it became activated when these types of virus secondly enters into the body. This is the classical mechanism of immune response against COVID-19. And then, if this immune response becomes breakdown, so there will be an infection. So, for the infection, specifically, the coronavirus enters through our nasal epithelium. The alveolar epithelial cells are most susceptible to this virus because it contains the high level of ACE2 receptor and TMPRSS receptor. And SARS-CoV-2 entered using those receptors, and they activate the some innate receptors like TLRs 7 and 8, RIG1, MDA5, and they recognize the viral particles, and they potentiate and generate the kappa beta mediated signaling pathway, and which generate the antiviral pro-inflammatory TNF1 cytokines. These are the pro-inflammatory cytokines, and but in case of the viral mechanism, the antiviral cytokines, that is the interferon 1 cytokines generation is much lower, and that's why virus replication rate become gradually activated. And this type 1 interferon exhibited by the production of ISGs such as RNA-seq. This protein attracts monocytes, macrophages, and T lymphocytes. And in the infected area, there is the several types of immunocompetent cells, especially the monocyte, the macrophages, and the eosinophils. These are all inflammatory cells also, and they become overactivated and releases a huge number of the pro-inflammatory cytokines, including TNF alpha, interleukin 6, interleukin 1 beta, and MIP1, and these numerous cytokines form the cytokine storm. And if this cytokine storm occurs in the lungs, then the lungs tissue become greatly damaged. And if this cytokine storm migrate towards the other organ, then this causes this organ specific damage. So there are various types of vaccines against the viral infection. Some of the vaccines are inactivated vaccines. Some of the live attenuated vaccines where the live pathogens, enters into the body, and their pathogenicity should be completely demolished, or otherwise they can generate the pathological consequences in the body. Some of the protein subunit vaccine, where there's a small part of the virus genetic, small part of the proteins, and the code of the proteins are entered into the viral genetic codes, and this virus, when enters into the body, they can express such proteins, and these proteins can be taken up by the antigen presenting cells, and by the conventional T cell B cell cooperation system they can generate numerous antibody against those viral proteins. And some vaccines are also known as the nucleic acid vaccine, whereas the DNA is injected into the cells for synthesizing target protein. Some proteins are the viral vector protein, but recombinant DNA technology is used for generate viral vector vaccines. So in India, I have already told that two vaccines, that is the Covaxin and Covishield, are currently used. And Bharat Biotech International Company in Bangalore, and with the collaboration with the Indian Council of Medical Research and National Institute of Virology, first approved COVID-19 vaccine, which is named the Covaxin. It is a purely indigenous vaccine. And another vaccine, that is the Covishield vaccine, which has been developed by the Serum Institute of Pune based on the AstraZeneca model. And this vaccine is basically a recombinant type vaccines where the spike proteins, the code for the spike proteins is given into the non-replicating adenovirus, and this virus expresses this spike proteins towards immune competent cells in our body. But both types of vaccines stored at 2 to 8 degrees centigrade, and both vaccine needed the 2 dose. But here, some of the recent research suggesting that not only 2 doses is enough for the successful vaccination, because it's a retroviral, and retrovirus have the tendency to spontaneously mutate, and their surface protein, their integral protein also. So that's why the longevity of the memory B cells is not giving the protection against the retroviral disease. So it needs a booster dose to obtain the maximum protection against those viral. So in Covaxin, aluminum hydroxide based adjuvant is used. We know that what is adjuvant. Adjuvant is a certain kind of substances which basically enhance the immunity of the antigen. So it is aluminum adjuvant or aluminum hydroxide adjuvant is one of the most widely used adjuvant, which has been taken due to its higher stability and effective potency for the vaccine formulation. Basically, there are immune response against Covaxin. Basically, the Covaxin potentiate the humoral immunity, but not usually the cell-mediated immunity. But certain kinds of the cell-mediated immunity also observed in some of the studies. Antigen-presenting cells mainly represent those digested antigen using the MSC class 1, class 2 molecules for the generation of the cytotoxic lymphocyte and T helper cell. These cytotoxic lymphocytes are become ready to kill the virus using their classical mechanism, which is mediated to the Fas and Fas-ligand interaction and release of enzymes, release of perforin, and calcium influx. Whereas CD4 T lymphocytes are mainly classical cells for generating the antibody response and also to differentiate into the plasma cells and to generate the memory cells. So this T helper cell 1 acts in the cell-mediated immunity and release cytokines, interferon gamma, interleukin 2, and TNF alpha, which further activates the macrophages, cytotoxic T lymphocytes, and T lymphocyte also eliminate the viral, also helps to eliminate the viral load in a very short period of time, because antibody generation of the antibody takes some time for seroconversion. It generally takes 1 to 2 weeks for complete generation of the antibody. But whereas when the cytotoxic lymphocyte become formed or become ready to infect the virus, it can ready to destroy the virus. It can destroy a large number of the virus within a very short period of the times, because they are due to their distinct mechanism of action. So this is the classical representation of the immunological response of Covaxin and Covishield. So in case of the Covishield, I have already told it is a viral vector vaccine where a non-replicating adenovirus is used, and this virus basically helps to synthesize the spike protein of the SARS-CoV-2. And these spike proteins are recognized by the several immunocompetent cells, And there are also the same types of cytotoxic T cell generation and CD4 T cell generation occurs in the same manner like the co-vaccines. The basic difference is the formulation of the virus. The easiest formulation of virus has been opted for the co-vaccine, and more modern formulation has been opted for the Covishield. And now, being this is it is a RNA virus, and so that it undergoes various types of mutation. This mutation basically based on the environmental challenges, individual to individual challenges, basically on the geographical location, based on their geographical location also, and using this such combination of such cumulative factors, the various types of variant strains have been developed, and this has been also recognized by the World Health Organization, where alpha strain has been denoted for the B1117. This is also a UK variant, whereas the beta strain, beta variant is known as the B1.351, which has been found in the South Africa, and the gamma for the P.1, which has been found in the Brazil, and delta strain for B1.617.2 is found in India. Now, delta plus strain also has been discovered in some parts of India, which is also alarming. Because the existing vaccination may not work properly in case of the delta plus strain, and it has been already reported that in case of the delta and delta plus strains, the efficacy of the both vaccine, that is the Covishield and co-vaccine, has been already decreased. The known mutation of COVID mutation, which has been found in the double mutation in India, is a combination of the two or more variants found in the Punjab, Delhi, Maharashtra, and classified at the P1.617 variant, which contains L45-2R and ED84Q variants. This is specifically the types of mutations which is found in the Indian variants. And COVID-19 triple mutant strains also found in some parts of the Maharashtra and Delhi during the second wave are more complex, because these types of variants show very much infectivity patterns, because their infectivity rates are more deadly than the classical infectivity rates of the SARS-CoV-2. The B1.617.2 variants, also given the name of the Delta variants, it is highly contagious, and it has been initially found in the India in December 2020. And the Delta plus variants also an additional mutation in the Delta variants, which is called the Ka41.7N on the coronavirus spike proteins, along with the existing mutation found in the beta gamma variants. That means the Delta plus variants is the combination of the various mutation. This is the combination of the various types of mutation, which consist of the alpha, which consists of the beta, which consists of the delta. And it is firstly found in the South Africa, Brazil, and respectively. And due to such mutation, this delta plus strains is very much contagious, very much deadly. So, when we consider comparative therapeutic advantage between the co-vaccine and Covishield, we can found based on their type-oriented differences, which I have already discussed. And their efficacy rate of the co-vaccine has been found 70 to 80%, whereas Covishield can be 90% effective. There are various research articles which support that the Covishield showing better efficacy than co-vaccine. Why it is showing, I will discuss in next slides. Antibody response after second dose of co-vaccine, the level of IgG anti-spike antibody decreased, and the co-vaccine has the lower efficacy after the first dose than the Covishield, whereas co-vaccine produces 80% antibody in body. In case of Covishield, single dose of Covishield, the amount of anti-spike IgG antibodies increase, which the peak has been observed after the 28 days of administration. Covishield produces binding antibody immune reactivity by increasing antibody against the spike protein in most people after 21 days after the first dose, but the Covishield can produce 98.1% of the antibody in human. This also obtained from some previously published reports. And when we consider about the side effects, so localized side effects of the Covaxin include pain, swelling. These are the very minimum side effects which can be treated using the very common drug like paracetamol, acetoprenate, etc. And in case of the Covishield, there are also some localized side effects, which is very common for every type of vaccination. So there is, in case of side effect, there is no such differences. There are some limitation. That means who can take the Covishield or who can take the Covaxin. And in case of the Covishield, the same condition applies at the Covishield. That means the if a person are allergic to vaccine ingredients or allergic to or already are skin allergy. There are several types of skin allergy are present, or suffering from any deadly disorders, etc. So that person, in that cases, these persons are advised not to take the vaccines. The level of the titer antibodies at a 51 unit per ml has been reported by the Singh et al. In 2021. Yeah, in case of the Covishield, it is 115 units per ml. So titer is also, there is much difference between the titers. Hello. So there are various types of vaccines which are the upcoming vaccines in India, and because India is a highly populated country, the classical ways of vaccine administration is not 100% showing 100% success rate, because the clinical setups, clinical setups which required for the vaccination program, to run the vaccination program, is not available in entire country. Moreover, the lack of the clinical practitioners, health workers are the other major types of problems. If the natural vaccines or some types of spray-like vaccines came in the future, so it will be very much helpful to cover the entire populations within a very short periods of time, because it can be the patient can easily take them using their nasal drops or nasal spray. And some of the upcoming vaccine like Sputnik 5, which has been already administered in some parts of this country, a biological E vaccine, also called Coblivax, which is a similar to Novavax vaccine. And the upcoming vaccine is also an upcoming vaccine in India. It's also required the 2 dose, and it also a type of protein subunit vaccine. Zycov-D vaccine is also the DNA vaccine. It's also against the major viral membrane proteins, not only for the spike proteins, also effective against the major viral membrane protein. Nasal spray vaccine, which I have just talked about, that it may potentiate the current vaccination program to various degree, because the administration issue is very much simple here, and there are no required to use the syringe like other types of vaccines, and people can immediately administer those vaccines. So as till now, there is no significant mutation, alteration in the spike protein has been noted. This is the very good news for us, because both two vaccines, that is the Covishield and Covaxin, are two vaccine based mainly generate the anti-spike antibody. But there is major difference because that Covishield only can generate the anti-spike antibody. But in case of Covaxin, the Covaxin is given with the whole virion, inactivated virion virus particle has been given to the body, combining the aluminum hydroxide adjuvant. So when the body is recepting the full viral particle, so it is not only generating the anti-spike antibody, but also the body's immune cells can generate various types of antibody, which is based on the viral surface protein, viral integral proteins also. So a variety and diversified antibody is generated in case of the indigenous Covaxin. So, if there is ongoing spontaneous mutation in the spike proteins of this virus, so in some cases this Covishield may not work properly. So in that cases, we have only our hand, that is the Covaxin, because it contains, it can generate multiple epitopes, and it can generate, and thereby it can generate multiple types of the antibody. So some types of the antibody will be effective to eliminate those virus particles if the significant alteration occurs in the spike protein. So some of the studies and some of the clinical trial already undertaken by the ICM are combining those two vaccines, that is the Covishield and Covaxin, one dose of Covaxin and one dose of Covishield, and vice versa. They want to study by mixing the vaccination doses, by mixing the vaccination types. They initially reported the efficacy rate become very much high. But they are concerning about the slightly high cytotoxic effects or side effects. But they think those side effects also can be managed using the available drugs. So from this study, we found that till today, the effectiveness of the Covishield is much higher than Covaxin. Why it is much higher? Because Covishield is only containing the spike protein materials, which after entering into the cell, the adenovirus expresses those spike proteins, and our immunocompetent cells, specifically the humoral immunity, become activated, and this generate specific antibody against the spike protein. So in immunology, it is very well known that when the single types of antigen or single types of immunogen challenge in our body, the immunity becomes superior. But when there will be the mixture of antibody, the specificity will be lower, but diversity will be higher. So in that case, Covaxin is better. But the everything that are related with the efficacy of the Covaxin and Covishield depends on the mutation rates. So if there is a higher mutation, or there is a significant mutation which significantly alters the epitope structure or the protein structure of the spike protein, then this efficacy rate will be decreased surely. And another important fact is that due to the large population, we need the simplest form of the vaccine, like using the nasal drop or some types of oral vaccine if possible, to cover up the entire population within a very short period of the time. These are the differences. And I want to share some idea or some knowledge. This is for specifically for the students, that what is the scopes of the immunology, studying immunology, or to form immunology or to take immunology as a career perspectives. So for their job profile, there are they can prepare themselves for the researcher. They can be a research assistant. They can be a lecturer in the various academic organization. They can be the therapist, and also they can be the consultant, and they can also work with the healthcare organizations, and sometimes they also create their own startups. So immunologists have the very distinct jobs, and they can examine the patient along with the clinicians, and they can diagnose or treat a variety of immune disorders, and they can also perform various types of experiments, studies, examine the results, discuss them, and also they can hypothesize new types of way out, or they can so for immunology courses, there are various specialization available, which are the cancer immunology, diagnostic immunology, microbiology immunology, and medical microbiology immunology, reproductive immunology, clinical immunology, evolutionary immunology, allergic and immunology and blood transfusion. So there are various types of courses, specialization courses, which can be obtained during the master degree level study. When a student want to pursue his master's degree in the immunology, they can take this type of specialization, which is offered by the various types of institution. There are various. This is the example of some top university who is offering the immunology courses in India. First one is IISc Bangalore, one of the very well reputed institutes. Jawaharlal Institute of Postgraduate Medicine, Education and Research, JIMPA, and Sanjay Gandhi Postgraduate International Medical Studies, National Institute of Immunology, MGR University, MIT University, and various very reputed university in India which are offering immunology at the courses for master degree level as well as for the BSc level. This is the course duration, which is same like about our general courses in physiology or microbiology. And doctoral degree courses can be done in the medicine in clinical immunology, philosophy of immunology, philosophy in cancer immunology, etc. So thank you. That's all. If there is question, you may ask.
Thank you, sir. We have so many doubts and queries and questions also about the vaccines. So I think everyone will be benefited from your talk. There are two questions, I think. One is Aparna Dash. Aparna, you have raised your hand. Now you may say and feel free to communicate with Dr. Sandeep Kumar Dash if you have any question. Please on your microphone and communicate with us. Aparna, if you have any question.
হ্যাঁ স্যার, আপনি বললেন যে কিছু কিছু ক্ষেত্রে কোভ্যাক্সিনের সেল মেডিয়েটেড ইমিউনিটি দেখা গেছে, তো সেটা কিভাবে?
কোভ্যাক্সিনের ক্ষেত্রে সেল মিডিয়েটেড ইমিউনিটি তো দেখো, যখন মানে মেনলি যখন ধরো তোমার কোনো ভাইরাল পার্টিকেলকে শরীরের মধ্যে ঢোকানো হয়, সেটা ইনঅ্যাক্টিভেটেড হোক বা অ্যাক্টিভেটেড হোক, তো ভাইরাসের এগেনস্টে একটা কি হয় না, ইনিশিয়ালি মানে সাইটোটক্সিক টি সেল রেসপন্সটা গড়ে ওঠে। কারণ সাইটোটক্সিক রেসপন্সটাই হচ্ছে ইনিশিয়াল ওয়েতে কিন্তু ভাইরাস কিলিং এর জন্য মেনলি ইম্পর্টেন্ট। এবার আস্তে আস্তে কি হয় না, টোটাল ইমিউন রেসপন্সটা কিছুটা সাইটোটক্সিক মানে CD8 রেস্ট্রিক্টেড, কিছুটা আবার CD4 রেস্ট্রিক্টেড চলে যায়। CD4 রেস্ট্রিক্টেড যে ইমিউন রেসপন্সটা হুইচ ইজ নোন ফর দ্য জেনারেশন অফ দ্য স্পেসিফিক অ্যান্টিবডি। সো মানে এটা কিন্তু এই যেহেতু কোভ্যাক্সটা দেওয়া হচ্ছে, কোভ্যাক্সিনের ক্ষেত্রে কিন্তু মেনলি এটা ব্যাপারটা আবার বেশি হয়, কারণ সেখানে হোল ভাইরাল পার্টিকেলটা দেওয়া হচ্ছে বলে। এই কারণেই আর কি মেনলি এই জিনিসগুলো দেখা যায়। তো স্পেসিফিক্যালি দুটো ইমিউনিটি চলে, কিন্তু মেজরিটি ক্ষেত্রে ভ্যাকসিনের ক্ষেত্রে টোটাল ইমিউনিটিটা কিছুদিন পর কিন্তু পোটেনশিয়েট টুয়ার্ড দ্য হিউমারাল ইমিউনিটি অনলি।
আচ্ছা স্যার।
Thank you, sir. The next question from Tanushree Mondal. Sir, why allergic persons cannot take the vaccines?
আচ্ছা, অ্যালার্জিক পার্সনের ক্ষেত্রে যেটা ভ্যাকসিনের এখনো পর্যন্ত যে এই ধরনের কোনো ট্রায়াল গুলো হয়ে ওঠেনি যে অ্যালার্জিক ইন্ডিভিজুয়ালসদের উপর ভ্যাকসিন দিয়ে কোনো রকম অ্যাডভার্স বা সিভিয়ার আবার অ্যালার্জিক রিয়াকশনটা বেড়ে গেছে বা অন্য কোনো কমপ্লিকেশন হয়েছে কিনা, সেটা এক ধরনের কারণ। আর দ্বিতীয় কারণ যেটা আমার মনে হতে পারে, ভ্যাকসিনের যে কম্পোজিশন গুলো থাকে, যেমন ধরো অ্যাডজুভেন্ট ইউজ করা হয়, স্টেবিলাইজিং এজেন্ট হিসাবে কিছু জিনিস ইউজ করা হয়, সেই জিনিসগুলো অনেক সময় কিন্তু লোকালাইজ অ্যালার্জিক রিয়াকশন তৈরি করে। এবার যেই পার্সনদের ক্ষেত্রে এই ধরনের অ্যালার্জিক প্রি এক্সিস্টিং ম্যানারে থাকে, তাদের ক্ষেত্রে কিন্তু এই জিনিসগুলো হলে কিন্তু সেই ব্যাপারগুলো আরো মানে খুব মারাত্মক আকার নিতে পারে। আর স্পুটনিক 5 এর যে মেকানিজমটা হচ্ছে, যে স্পুটনিক 5 হচ্ছে রিসেন্টলি এটা স্টার্ট হয়েছে, এটাও এক ধরনের তোমার হার্মলেস ভ্যাকসিন। মানে মেনলি যেটা হচ্ছে তোমার যে ধরনের আমরা নিচ্ছি, যেমন কোভিশিল্ড নিচ্ছি, সেই টাইপের খানিকটা অনেকটা এই ধরনের মেকানিজমের উপর বেস করে রয়েছে স্পুটনিক 5টা। মেনলি এটা তো তোমরা জানো যে রাশিয়াতে ডেভেলপ হয়েছে। ভ্যাকসিনটা, কিন্তু সেই অর্থে এখনো পর্যন্ত যে ডেটাগুলো আমরা যেমন ধরো কোভিশিল্ড, কোভ্যাক্সিনের ইন্ডিয়ান পপুলেশনের উপর অনেক ডেটাগুলো পেয়েছি স্পুটনিক 5 হিসাবে, কিন্তু সেই টাইপের ডেটাগুলো এখনো উঠে আসেনি। তো তাদের এক্সাক্ট কি ধরনের ইমিউনোলজিক্যাল রোলস বা ইমিউনোলজিক্যাল কি পোটেন্সি বা সাইড এফেক্টস সেগুলো কি ধরনের হচ্ছে, সেই ব্যাপারগুলো কিন্তু এখনো পর্যন্ত সঠিকভাবে জানা যাচ্ছে না, যে বডির মধ্যে তারা কতটা পরিমাণে অ্যান্টিবডি তৈরি করছে। বাইরে ওরা ক্লেম করছে যে মোর দেন 90%, কিন্তু ইন্ডিয়ান পপুলেশনে কত, কারণ ইন্ডিয়ানদের ক্ষেত্রে দেখবে যে এই ভেরিয়েশনটা অনেকটাই ভ্যারি করে ওয়ার্ল্ডের অন্যান্য কান্ট্রির তুলনায়। কারণ অনেকে বলে ইন্ডিয়ানরা হচ্ছে ইমিউনিটির দিক থেকে অনেকটা বেশি স্ট্রং, কারণ আমাদের যে অ্যাটমসফিয়ারে আমরা বসবাস করি ছোট থেকে ধরো যেখানে গড়ে উঠি, সেটা ধরো অনেক মাইক্রোবিয়াল চ্যালেঞ্জ আমাদের সাথে হয়ে থাকে। সেই চ্যালেঞ্জের ফলে কিন্তু বডিটা আস্তে আস্তে কিন্তু তার এগেনস্টে রেজিস্ট করতে থাকে এবং তার এগেনস্টে কিন্তু প্রোটেকশন নিতে নিতে এমন একটা জায়গাতে চলে আসে যেখানে অন্যান্য কান্ট্রি, যেখানে ধরো যাদের ক্ষেত্রে যেমন ধরো আমেরিকা যদি ধরো, এদের তুলনায় কিন্তু দেখা গেছে যে ইন্ডিয়ান পপুলেশনের মধ্যে ইমিউনিটিটা অনেকটা বেশি। তো সেই কারণে হচ্ছে ইন্ডিয়ান পপুলেশনের ক্ষেত্রে এই ধরনের ভ্যাকসিনেশন ট্রায়াল গুলো সবসময় আমরা ওয়ার্ল্ডের রেসপেক্টে ধরে নিই না, কারণ ইন্ডিয়াতে এদের এফিকাসি রেটটা অনেকটাই চেঞ্জ হয়েছে। আগেও দেখা গেছে অন্যান্য ভ্যাকসিনের ক্ষেত্রে এই রেটটা অনেকখানি চেঞ্জ।
There is another question from Tanushree. Is there any complication for blood donation after vaccination?
ব্লাড ডোনেশন না, কোনো
I will share. It is visible.
Yes.
Visible and audible also.
Okay. So yeah, I think So the student maybe they are overburdened, back-to-back 3 serious discussion. So believe me, I will not overburden with my presentation. So I'll give a very informative presentation that will help for their career. And I need, actually I will not say it's a deliver thing, better I will say it is an interaction. So I like to active interact with the student. So my topic is ergonomics, a great career opportunity for design mindful student. So when I was in my graduation or post-graduation stage, so like me, so it's obvious for all people, all the student. So what do you do after your graduation or post-graduation? So maybe they'll be confused. They might go for school service commission or public service commission. They might go for college service commission or research also. But that is a very well-known area where you can work, and definitely they are mind-blowing. But if you think something different, then I have one suggestion. So you can pursue your career in design. So you might be surprised that you are the student of physiology, means human physiology, and how you can pursue your career in design. So the first thing, when I was maybe in the graduation stage, so that time even I also feel the design, that means creating some craft or something is the design. But there is more than that. It's a very small part of that. So now you are the student of physiology. So how you can link with design? This is the bridge is ergonomics. So ergonomics, it's a subject. You can do your master, you can read ergonomics as a special paper from human physiology. So the first thing, what is ergonomics? Though I will not go detail about that thing because it's actually I'm not delivering on the ergonomics. I'm not teaching you. So if you have interest, definitely you will learn it. So just I'm giving a very basic idea. The ergonomics is a science that dealing with designing a safer and efficient environment for maximum productivity with minimum human cost. Some other people also define ergonomics like that. Ergonomics is a science of fitting the job to the worker when there is a mismatch between the physical requirements of the job and the physical capacity of the worker. So maybe it going above your head in that stage. So what I will say, so ergonomics helps people to do the job as their own way. The people, all people, they have some strength and also some limitation. So ergonomics helps to continue the work on his strength path, strength portion, and avoid the limitation. So the goal of ergonomics is to make the work fit the worker instead of making the worker fit the work. So I'll explain this, understand the strengths and limits of the user. User means the people who will use a product. So they have some strengths and also the limits. So you have to know which is both the thing, the strengths and limit of a user. I'll explain this from the history of ergonomics. So during the World War II, so there was some accidents happened for the fighter planes. So then they try to figure it out why that is happening. So it is a pilot error or it is a design fault. Then it came a new subject, ergonomics. And the reason of this crash, I will explain in the next slide. And definitely, it was not the pilot error, it was the design fault. So there was two gears for the landing gear and the landing flaps. So in the initial design, during the World War II, the firecrafts, they have the same gear, the landing gear and the landing flaps. But during the emergency, people or user do from their instinct. There is no no lesson or no training will act there in that particular situation. So how to avoid this problem? Yes, just change the gears design. So there will be two separate design. So by looking this, the user can identify, okay, this for the landing gear and this is a landing flap. So human error might be removed. So another problem I'll discuss here. So it's about the door. I think many of you found difficulties with this kind of problem. So maybe you are standing in front of a glass door, then it will come in your mind, you have to pull or push. I think it happens to everybody. But the door is very simple thing. So the work of door is just to allow a person, it can open and close. Only two options is there. But still, actually it's the opening or closing door is not a rocket science. It's very simple activity. Still, people there are some error. So is this human's error? No, it's not the human error. It's the design fault. So how we can solve this issue? It's very simple. We can use just from the image, you can see there are two things are there, push and pull. If you use the pull plate, there are no handle is there. So when you have a look the image or the product itself, and immediately you can find, oh yes, I have to push. But when the handle is there, so immediately you can understand, oh, I have to pull. I'm giving another ergonomic issues. I have taken this image from my room. So it's a switch board. So there are three switches that I marked with 1, 2, 3. And I'm giving hints that these three switches for the light, fan, and flag point. So if I ask you, so which switch is for the which purpose? I think all of them, means all of you, immediately say that the switch number 1 is for the fan, switch number 2 for the light, and switch number 3 for the plug. How you can understand this? Because there is a fan regulator is there. So just beside the regulator, the switch is placed. So it must be the switch of fan. And on the right side, there is a plug point, and just beside the plug point, the switch is there. So it must be the plug point, and the rest will be the light. So if you means this called stereotype. So when you design something, you have to concern about the human stereotype. So this stereotype might be different from area to area. If you compare with the Indian scenario with the UK or US, there will be some lacunae. So there must be some mismatch. So when you are designing something, so you have to know the user. That's the first thing. So I won't go more details about the stereotype. I think Dr. Mona Lisa, she can clear this thing better way, as she has done her PhD on that topic. So next is ergonomics is a career opportunity. So designers are everywhere. There is no life without a designer. I'm giving one example. In the morning, you get up and maybe you are using a toothpaste and toothbrush. So both the things are product, and someone designed it. So is designed is not only the making the craft, design also to make a product, that's also a design. So in each and every step in your life, in your daily routine, you can't avoid the design. So designer is everywhere. So design is a way. So after completing your course in ergonomics, where you can work. You can work in a textile and fashion design industry. You can work animation design industry, film and video design, graphic design, furniture design, interior design, product design, mobility design, and many more. So each and every company needs a designer as well as ergonomist. So I'll say sky is your only limit, because the designer opportunity is there in every place. So I have placed these 3 screenshots that I have taken today morning. Just I searched there in the Google, the ergonomics job or human factors job and human factor engineering job. So if you search with these keywords, you will find the lots of advertisement. They need the human factors or ergonomist. So yeah, one thing I will clarify. So ergonomics and human factor specialist, human factor is both is the same term. So in Indian continent, we call this ergonomics, but maybe some other area like US and Canada or UK, they call as a human factor. So ergonomics and human factors both are same. So for searching a job, maybe you can use all 3 keywords. So next, what you need to work as an ergonomist? So definitely you need the knowledge of ergonomics. So all products that's made for the human, and you are the physiologist, you know the human better way. So there should be a match between the design and also the user, like human. So you are a great person to match the design with the user. Then is a basic idea of design process. Definitely it's not, you will not read this thing in your ergonomics course, but if you want to pursue your career in ergonomics or human factor engineer or human factor specialist, definitely you have to learn the basic idea on the design process. You have to be design mindful. So what is design mindful? I will tell in later stage. You have to confident to work in a team, because when you design something, you won't be only one person. You will be one resource person from the ergonomics, but there will be some other person from the mechanical engineering. There will be some other person from industrial design department. So you have to work together. And yes, you have to learn some 3D software like AutoCAD, CATIA, SolidWorks, and many more. So I'm giving one just example. So that screenshot I took from CATIA. So CATIA is a software where you can build a human mannequin. So here you can put some dimension, and accordingly you can make the mannequin. Then what you will do with the mannequin? You can do the job simulation. So just for example here, I'm giving one example. So it's a chair, this chair made for the train industry for the passenger. And here we are doing some simulation about the footrest location. So we place the, I think she is the 50th percentile mannequin on the chair, and we are doing some study here, and we are fixing the food test position. So this CATIA, the one very familiar software in design industry. And the another thing you need, the out of the box thinking. So in design language, it's called lateral thinking. So what is lateral thinking? So maybe we'll do one or two experiments. So can you unite the below 9 dots with 4 straight lines? You cannot put your pen before end. So you have to finish it, and then you can just end the, you can put out your pen. So it will be a continuous line. So can you please do it? Maybe I'll wait for maybe 30 seconds. You don't need to send here, but maybe you can just on your mic and you can say if you have something.
আমি পারছি।
Okay. Okay, so I'm going to there's a next screen. I think it will give you, there's a one line, the second line, it's third line, it's fourth line. So when you got this problem, so you are focusing only the 9 dots. So you are not going the outside. I have not said that the straight line must be within this box. So that's a lateral thinking. So you have to think beyond the limit, beyond the box. So we'll do another example, another exercise. Can you put below 4 dots such a way that the distance between each other would be same? I'm giving 30 seconds. And if someone can click it, then just on your mic and just respond. Anyone? Okay, so I'm going to the next thing. So this one. So here you can put this 3 dot in 3 corner of this triangle. So the distance will be same. It is called, I think, all sides equal triangle. But the fourth one, so right now you are thinking on the 2D. Just you think about the 3D. So there is one more layer on the top. So you can place the fourth one on that position and you can make it. So this called lateral thinking. So to be a designer, you have to think some different way. Then maybe you will get some extraordinary result, and you can surprise the other people or maybe your client, your customer, with the new design. So now I'll explain the design process, then I'll explain about the design mindful. So what is the design process? So as I'm working here in Bombardier Transportation, so Bombardier Transportation is a giant rail making industry. It is globally very famous companies who make the railways. So I'll give an example from that and I will explain the design process. Though it will take some longer time, but I made it very soft, very crisp and clear. So if you have any problem to understand that thing, please shout. The first thing, define the problem or analyze the brief. So the design process starts from this point. So either define the problem, maybe you are facing some difficulties due to some design, and that's why you want to change it or you want to redesign it. Or the another option, maybe the client came to you and asked, okay, something that is there in their mind and they want to design something. So I'm giving one example. So in UK, there is a railway franchisee called SWR, Southwestern Railway. Actually, in UK, the railway is a privatized. It's not the government thing. So the Southwestern Railway, they came to us, means Bombardier Transportation, and they asking for a cab, to design a cab, and they gave 174 of controls. So you have to place this number of controls in your cab and you have to design it. So that is the first stage, that information or approach we got from the client, or we understood the problem. So what is the problem here? So we have to put all 174 controls such a way so that can be used by the user. The next step is identify the key requirement. So what is the requirement? So the customer or client, they asked for the ergonomic cab for the 95th to 55th percentile UK driver. So I believe that all you know about the percentile. So the 95th to 5th percentile people, so you all have idea about it. The next is the research ideas or explore the possibilities. So you got the problem, you got the requirement, then maybe you need some information. So you have to do some research. So maybe what the thing, the function, we got the 174 controls. So what the function of that control? The frequency. So the frequency of to use that control, the criticality of control. The criticality means how critical the control is for the driving activity. Then is the user body dimension. Okay. And next, develop mannequin. So you have to information. So that's called research. If for somebody maybe previous ongoing product, how they designed it, so it will also help you. So this one screenshot from establish the criteria and the constraints and limitations. Definitely, some controls within that area. But it is obvious that you cannot put all 174 controls in your primary zone. Definitely you have to use some other zone. Okay, so next, consider the alternative thinking, alternative solution that I just explained few slides ago, the lateral thinking. So here, what is my approach? So can I put some controls far from the body? Means instead of the that cab days, can I put some controls somewhere other areas, like maybe the cab back wall? Next one is the select some approach or idea generation. So here I'm giving one mind map. So what I did here, so there are 174 controls. So I divided in 8 groups. So one is the master control. When the driver is driving train, constantly he needs to hold the master control. So it's maintain the speed. The next one is the DMI. The full form of DMI is driver machine interface. So it is the interface between the driver and also the train. So the driver can visualize the speed, driver can visualize the train, the driving related status, the signal, everything on the DMI screen. The next is the TCMS. It's also the full form of TCMS is train control management screen. So it's also a touch screen. Here, the information related to train will be highlighted. The what information? It's not related to the driving. when you are driving a train, definitely there will be some saloon areas. So the passenger should be there. So the details about the saloons will be visualized here. Even people, you can see the lights, fans in the saloon, it's on or off, or maybe some other things also there. Then I figure it out the frequent control. During the driving, some controls are there. This control, the user needs to use very frequently. So that's a number 5. Number 4. Number 5 is the communication control. There are some controls that the driver needs to communication with the maybe the nearby station master or maybe the saloon passenger also. And the 6th one is the cab environment setup control. Some controls are there. That's are the set up the cab environments like the cab fan, cab light, something. The next is the CCTV cameras. So there are lots of cameras are there in the saloon. So the output of these cameras are here. That will be displayed here in CCTV screen. So the driver can constantly see what is happening in the saloon. And the number 8 is the isolation control. So there are some controls are there that's called isolation control. This control can be used not in the normal situation when the train is some abnormal situation. So the driver no need to reach that control during the driving. So what I did here, I did a very rough sketch here. So it's the human being. So this is the primary zone, this is secondary zone, and this is tertiary zone. So I place the master control here on the primary zone because the driver will interact with this master control each and every moment when the train is in running condition. The next is the DMI. So DMI I am keeping in front. So why I keep it in front? So DMI will display the status, is the screen, is a touch screen. It will display the train status, maybe the driving speed, and many more things. So I'm planning to place this thing in front of the driver because during the driving, he or she can visualize it and take decision. The third is the train control management system. That's also the screen. Even this screen also I am planning to put in the front, but bit left hand side. Then is the fourth is there are some controls are there, the frequent control. So I want to put the frequent control here. And there are some communication control that I like to put in the right hand side. And I want to put this isolation control on the back of the wall, cab back wall, because the driver doesn't need this control during the driving. So from that thing, you can make a model. So I told you, there are many softwares are there like CATIA, SolidWorks, Rhinos, many more, Alias. So you can take any one of them and you can make it. These are the model. So we made this model. So it's not the actual one. So it's called the render image. So it looks like actual photograph, but it is a model. So here, so I told you these are the DMI screen, these are the TCMS screen, and there are few controls are there, communication control on the right hand side, and these are the master control that driver needs to interact with each and every second while driving, and these are the some frequent controls, and these two are the CCTV cameras, the output of CCTV cameras. So the person need to look the cameras, but they don't need to reach it. So that's why it's a bit far. So there are some control that we placed regarding the cab environment setup, like the cab fan, light, everything. So the person can set up the cab and can sit here and drive here. So he or she doesn't need to interact with this control while the driving. And special special care was taken to put the emergency brake. So emergency brake is a very important control for any running design, like car or train, whatever. So the best place to keep the emergency control is in front of the driver, so that he or she can look the control each and every time, but it will be with maybe the secondary zone, not the primary zone. The reason is otherwise it can be accidentally, it can actuate it accidentally. So it's kept the emergency brake control, it was placed here. And there will be a second emergency brake control. So in train, there will be two people. One is the driver and second is the co-driver or second person. So though the second person will not drive the train, but still there is one emergency control placed near to the this area where the second person also can interact with this control. Maybe some situation means emergency, this brake with this control is required for the emergency purpose. And the second person also can use this. So next stage is to make a model or prototype. So to test it, here we made this prototype. So it's not the actual train. It's made with it's a wooden mock-up, 1:1 ratio. So here, these controls are not operating, just we place the low fidelity paper on that. So it's just we took the printout of the control and we are placing here. So it will give some views, some authenticity. Okay, you are sitting in a maybe real cab and you can do some testing. The next is test and evaluate. So where you will test it. So as per Bombardier protocol, they will make a full running train. It's in design language it's called unit 1. So unit 1 is the actual train, but it's not the production. But it is real. This train is made only for one to test everything, each and everything. So here means it's a original train, but it's not the production. Even still that time we have that option to change something in our design. So if something is wrong, then the next step is refine the design. If something from the testing of the mock-up and as well as the unit 1, if we found something difficulties, then we can do it, means we can change it. We refine it and we make the final design. After that, the design should go to the production. So now it was there in hand of the R&D, research and development. Now the design is ready for the multiple production and it should go to the production. And design, it's a continuous way, it's a circle. If something happens, even after releasing the train, even the same way it can move from starting to end. So that's why it is a circle. So this is a design process. Though there are many things are there, but I made it a bit short because of the time. The final thing is, you might be asked, am I design mindful? Because I said that ergonomics is a great career opportunity for design mindful student. So definitely now you might be interested to know, am I design mindful? The answer is yes and no, both. Why? The reason is all people are design mindful, but maybe it is there in your inside as a sleeping condition. So maybe you are not aware about that thing. So that's why, okay, you are just, maybe you are facing this issue and you are just ignore it. But so what you need some trigger. So that trigger will help you to understand that, to identify the design problem, and maybe you will think on that and you can make a good design. So I'm giving some example, some interesting thing about the design, though it is not the design class. So I'm just giving a interesting thing. So here you can see these two diagram, there is two circles there, A and B. So can you please tell me which one is the bigger? You can just on your mic and you can answer. Can you hear me?
Same.
Yeah, it is same. So now I'm giving one interesting fact about the design. It's called golden ratio. So what is golden ratio? So golden ratio, golden ratio is a special number and approximately equal to 1.618. What is that? So there is one line is there. So this part is A and this part is B. And the total length is A plus B. So each and every time A by B, A means the bigger part, A by B is the bigger part by shorter part equal to the whole length, like A plus B by the bigger part. And each and every time the value will be 1.618. You can put anything, means you can do the test, means you can take whatever you want to take it, and you can just do that calculation and you will get it this value 1.618. So this value is very important for designer. So that's why even it's called the nature number. Why? I'll tell you later. So from that thing, you can, this is a golden ratio, and you can get the golden rectangle. What is golden rectangle? So this is 1 unit and this is 1.618 unit. So it will give you a rectangle that is called golden rectangle. So the many designers use this thing, that ratio for their many purpose. I will explain later. So how to create a golden rectangle? Okay, so first you just make a square of unit 1. So 1 is there is a square, it's not the rectangle, and you place another square in the same value 1 side by side, and you will get some area, and you just make this area as another square. So the whole thing will be a rectangle, and this is the golden rectangle. So I'll tell you the importance of the golden rectangle. So even the whole figure, this figure also a golden rectangle. So the interesting thing, if you just delete a square shape from a golden rectangle, the rest of the part will be another golden rectangle. So this is the whole rectangle, and I am just deleting that part, the 8. So the rest of the part is this. Even this is another golden rectangle. Even if I delete another square from that part, the 5 one, so you will get the rest of the part that will be create the another rectangle, that's also a golden rectangle. And if you delete the square from that part, you will get the another rectangle, that's also a golden rectangle. So each and every time, it's unlimited time, if you delete or remove a square part from this rectangle, the rest of the part will create another golden rectangle. So next I will tell you about the golden spiral. So okay, let's talk about the golden circle. So what is golden circle? So in each and every square, you can create a circle. That's a maximum, the bigger circle, that's called golden circle. So I will tell you the importance of golden circle later. The next is golden spiral. So if you just place this golden circle, the 1/4 part here and 1/4 part, means each circle the 1/4 part here, so you will get a spiral line. So that's called golden spiral. Even the importance of this golden rectangle, golden spiral, and golden circle, I will discuss here. So if you have just carefully notice in the nature, you will find this golden spiral each and everywhere in nature. Just it's available in the flower, it's available even during the tornado. You will find here even the cell, the samuk cell. So snails, you will get a golden spiral here also. So that's why it called a nature's number. And from the survey, it is found that if you are designing something with this proportion, it will be a eye soothing. So many designers use golden ratio just for the eye soothing purpose. And the end of the day, being a designer, you have to sell your design. So you have to convince your client. So if you just inform your client or customer that it's made with a proportion of golden circle, so they'll be happy. So this image, so this ratio, the golden ratio is available on the human body also. So this image, it was made by Leonardo da Vinci. So if you take the distance from the navel to leg, means foot, and the rest of the part, you will get the same ratio, 1.618. Even you will get the same golden ratio here on the arms. Even you will get this same ratio on the nose. So you will find the golden ratio in human body also. So that's why it called nature's number. Then maybe I might ask you, what do the Parthenon in Greece and the Vinci's Mona Lisa have in Can you hear me? I think
Yes.
I got a power cut. Yeah, I got a
Yes.
power cut, and that's why the internet. Yeah, actually, and I saw many people from each and every part of the world, even from India also. But I have not found any people from West Bengal, especially the Bengali people. And then I realized that maybe the student of the West Bengal, they are not aware about the another the opportunities are there. So that motivates me to talk on this topic so that maybe I can give some trigger to the people in your college, and maybe if one of them coming in the design field, then I think I'll be happy.
Thank you again, sir. Actually, we are privileged. I will request Dr. Mona Lisa Banerjee to give the vote of thanks to all of us.
Thank you, ma'am. I hope I'm audible.
Yes, you may proceed.
Okay. So good evening, everyone. We have reached to the end of this engaging and informative session. First of all, I would like to thank our eminent speakers today, Professor Nihar Ranjan Jana, Dr. Gautam Datto, Dr. Sandip Kumar Das, and Dr. Amar Kundu. Then I'm conveying my sincere thanks to the chief patron of this webinar, Dr. Manush Ranjan Bhuiya, and he's the president of the governing body, and Professor Tapan Kumar Datto, he's the principal, and he's the patron of this webinar. And I'm thankful for their constant support and encouragement. And then I must have to thank Professor Atanu Samanto for his support. Without him, it was not at all possible. And finally, my heartiest thanks to all the participants for joining us. Thank you all.
In good night, everybody. We are in making an end here. Thank you.